So today we are going to talk about some general concepts related with the gastrointestinal system motility, right? First of all, I will talk about the basic structure of the gastrointestinal system, which I have already discussed in detail in the video related with the histology of gastrointestinal system, right? Then I will go into detail of special features of smooth muscles of the gastrointestinal system. As you see in this diagram, that this is the wall of the GIT which I'm showing, but I have enlarged, especially its upper part. And here is the lumen, and this blue lining, this is a mucosa, epithelium of the mucosa. Here is lamina propria, and here is muscularis mucosae. So these three layers, this is mucosa of the gastrointestinal system, and here I'm showing submucosa, and this is muscularis externa with circular layer of muscles which is inside, and longitudinally oriented smooth muscles which are outside, and this is serosa, right? So this is the basic structure of gastrointestinal system. Now, in this gastrointestinal system, what we can see that smooth muscles are present as a very thin film in the muscularis mucosae in this area. Is that right? But most of the smooth muscles in gastrointestinal wall are present in, what is this layer? Muscularis externa. And these smooth muscles are mainly in two layers. The inner layer of smooth muscles, which are circular layer of the smooth muscle, and outer layer of the smooth muscles, which is basically longitudinally oriented. Now let's go into detail of smooth muscles of gastrointestinal system. Actually, smooth muscles of gastrointestinal system, they make large bundles, right? These bundles, right? These bundles, usually one bundle has about 1,000, right? Smooth muscle cells. Let's suppose that this is one bundle and it has at around 1,000 smooth muscle cells, right? And these bundles of the smooth muscles which are present in gastrointestinal system, right? These bundles are oriented longitudinally in the outer layer of muscularis externa, and same bundles are present circularly in the inner layer of muscularis externa. Now, within this bundle, what really happens that these smooth muscles are interconnected with each other through gap junctions, right? What I mean by gap junctions, let's suppose I make one smooth muscle here, and suppose here are multiple smooth muscles of gastrointestinal system. Now, actually, of course, these are the nuclei in the smooth muscle. Now, let us suppose these few smooth muscles represent one bundle, but of course there are in one bundle there are about 1,000 smooth muscle cells. These smooth muscles are connected with each other through gap junctions. What are these? These are gap junctions. And these gap junctions are low resistance communication systems. So you can say that one smooth muscle can communicate with the neighboring smooth muscles through these specialized gap junctions. Gap junctions are specialized areas where smooth muscles come in touch with each other, and smooth muscles are having special channels or special electrical windows or channels, low resistance channels, due to which ions and small molecular weight metabolites can move from one smooth muscle to the other smooth muscle, right? And these gap junctions, one smooth muscle has multiple gap junctions which connect that smooth muscle with the multiple neighboring smooth muscles, right? Now, the point which you need to understand is, because all these smooth muscles in a bundle are connected with each other through the gap junctions, right? Now, any electrical activity, any significant electrical activity in one smooth muscle will be communicated to the neighboring smooth muscles also, right? So what does it mean? It means that electrical activity in one smooth, if you stimulate one smooth muscle, that stimulation will go to the all smooth muscles in the same bundles. Is that right? For example, if there's action potential, you stimulate this smooth muscle, right? Then you have stimulated one smooth muscle, but connect it will the ions, right, or depolarizing ions or repolarizing ions. Those ions will be connecting to each all these smooth muscles through these gap junctions. What does it mean? That all the smooth muscles in one bundle are behaving as a network of smooth muscles which are electrically syncytium. What is this? Electrically they are interconnected. If you stimulate any one, if I stimulate this smooth muscle, all others will be also stimulated. Is that right? Then second point you need to understand that not only gap junctions are present in between all the smooth muscles which are in one bundle, it is also seen that actually smooth muscles in different bundles also are connected with each other. Let's suppose there is a smooth muscle here or smooth muscle here. Now this is a separate bundle. But the point which you need to understand is that not only in this bundle of smooth muscles, all smooth muscles are connected with each other through the gap junctions. Not only they are here connected with the gap junctions, but a very important point is that smooth muscle in one bundle are connected also to the smooth muscles of the other bundle through the gap junctions. Is that right? So it means if you stimulate any smooth muscle in one bundle, then adjacent bundles of the smooth muscles will be stimulated. What does it mean? If I stimulate electrically this smooth muscle, right, then what will happen? That for example, it is undergoing action potential. Then this action potential or electrical change will spread not only into all these smooth muscles in this bundle, but it will also communicate to the adjacent bundle also. So what does it mean? That smooth muscles in gastrointestinal system, they are connected with each other through gap junctions, and due to this reason, they act as electrical syncytium. Is that right? And due to this reason, these smooth muscles in gastrointestinal system, they are called unitary smooth muscles. What are they called? Unitary smooth muscles. What I mean by unitary smooth muscle, that all these smooth muscles, the millions and millions of smooth muscles which are connected through gap junctions, they act electrically as one network, an intercommunicating network electrically. Am I clear? Any question up to this? Right. Another thing, as I told you, these bundles, where every bundle has about 1,000 smooth muscles, these bundles of smooth muscles are longitudinally arranged in outer muscularis externa, and same bundles are arranged circularly in the inner muscularis externa. But the point which I want to highlight, even there are few, not much, but there are few connections between the smooth muscles present in outer longitudinal layer and inner circular layer as well, right? But more gap junctions are present in between the smooth muscle in longitudinal layer, and more bundled gap junctions are present in between the smooth muscles in circular layer. But there are very few interconnections or gap junctions between the smooth muscle bundles in outer longitudinal layer and inner circular layer. Do you have any question up to this? Right? After learning this thing, then the next point which you need to learn, now what is the normal electrical activity in the gastrointestinal smooth muscle? As you know, in the heart, there is a pacemaker. In the same way, in gastrointestinal smooth muscles, some special cells which are called, there are some special cells which can electrically stimulate the smooth muscles, and these cells are called interstitial cells of Kajal. Interstitial cells of Kajal. Now these interstitial cells of Kajal, they are present especially in between the longitudinal and circular layer, right? And these special cells can produce pacemaker activity. They can produce what? electrical fluctuating electrical activity. Let's suppose if there's a cell here and this is an interstitial cell of Kegel, this can produce, suppose these are 3 cells of interstitial cells of Kegel, they can undergo, their membranes can undergo electrical fluctuations, and due to the gap junctions, these electrical fluctuations will be communicated to the neighboring smooth muscles, and then that electrical wave will pass from one smooth muscle to the next, so on and so forth through the gap junctions. Now, the question is that what is the electrical activity in one smooth muscle? Now I'm going to draw one smooth, let's suppose this smooth muscle I enlarge over here and show you what are the electrical activities which goes into a smooth muscle, right? Pacemaker activities from where? Interstitial cells of Kegel, right? They basically produce the electrical activity which spreads to the neighboring smooth muscles of gastrointestinal system through the gap junctions, and then this electrical activity, depending upon the excitability of the smooth muscle, will spread to wider areas. Now let's see what is the electrical activity in one smooth muscle, right? Now I'm going to draw one smooth muscle. Actually, if you study the electrical changes or voltage changes in the across the membrane, right, of the cell membrane of the smooth muscle, you will find that the electrical potential inside the membrane fluctuates, right? And let me show you how are those fluctuations. Those fluctuations which are normally occur, and these fluctuations are called slow waves. Now let me explain what are slow waves. Now, let's suppose, now what is here, that this is suppose resting membrane potential. Now resting membrane potential in the smooth muscles of gastrointestinal system is somewhere between, yes, minus 60 millivolt, right? And here it is, suppose minus 40 millivolt, right? Now, what really happens in the smooth muscles of gastrointestinal system, there are special channels. There are 2 theories. One theory is that there are calcium channels and which allow the small amount of calcium to come in, right? And if small amount of calcium is trickling inside these smooth muscles, what will happen as cations are coming in, then resting membrane potential will move towards, yes, it will become less negative. What will happen? This potential, which is minus 60, it will become minus 55, it will become minus 50, even it may become minus 45. So what really happens, that there are special type of channels present in the membranes of gastrointestinal smooth muscle which allow a little bit sodium and calcium to come in, right? And slowly the resting membrane potential become less negative or become slightly depolarized, right? Slightly depolarized, but it will reach to the peak. Once it reaches to the peak, then what happens, that potassium channels open and potassium goes out. And when potassium is going out after the peak, as potassium channels open, right, what will happen? Potassium will start leaking out as positive ions are being lost, then progressively membrane will become more negative and reach to it resting membrane potential, minus 60. But as soon as it touches there, again, these sodium calcium channel allow again the sodium and calcium to trickle in. Then again, what happens, the resting membrane potential, due to incoming influx of calcium and sodium, it again start going upward, right, and become slightly depolarized, right? But as soon as it reaches to peak, what will happen? Again, potassium efflux will start and resting membrane potential will become again to its resting situation. But again, cycle will be repeated. Is that right? Due to these repetition of these cycles, we can say the membrane potential inside the cell membrane of the smooth muscle fluctuates. It becomes slightly depolarized, then repolarized, and slightly depolarized, then repolarized, slightly depolarized, then repolarized. And this pattern of fluctuation, that membranes slightly undergo depolarization, again repolarization, depolarization, again repolarization. These fluctuating potential changes in the membrane potential of smooth muscle are called slow waves. What are they called? These are, yes, slow waves. Remember, these slow waves are not action potential. These slow waves are not action potential, and these slow waves are inherent tendency of the gastrointestinal smooth muscles. Then again, these slow waves in the, what is this? Smooth muscles of stomach. In the smooth muscles of stomach, these slow waves are produced 3 times per minute. 3 times per minute. And same slow waves are produced in jejunum and ileum about, yes, 9 times per minute. And same slow waves are produced in the duodenum about 12 times per minute. Let me repeat it. In the stomach, these slow waves, which are undulating membrane potential, right, with repeated depolarization, repolarization, depolarization, repolarization, depolarization, repolarization, these slow waves are produced, right, in the stomach smooth muscles about how many times? 3 times per minute. In the duodenal smooth muscle, 12 times per minute. 12 times per minute in the duodenum. And in the jejunum and ileum, it is 9 times per minute. Am I clear? Now, actually, these slow waves are not action potential, right? And these slow waves do not produce contraction in the smooth muscle, right? They just set the basic frequency of electrical fluctuations. Now, what happens? The resting membrane potential is at -60 and threshold potential is -40, right? This is resting membrane potential, and what is here? This is threshold potential, and this is resting membrane potential, which is -60. So now usually, right, what happens? It means that when cations are coming in, right, resting membrane potential is approaching threshold. So smooth muscle is becoming more excitable. And then as potassium cations are going out, and after achieving the peak, when potassium efflux start, then potential is becoming more negative and cell is becoming less excitable. So what really happens, we can say in another way that these slow waves, right, when they are produced, then gradually what happens, that excitability of the smooth muscle increases progressively and then decreases progressively. Now, listen now very carefully, if this peak of the slow wave touches the threshold, if peak of the slow wave touches the threshold, then voltage sensitive calcium sodium channels will open. These voltage sensitive calcium sodium channels are different than these cationic channels, right? These cationic channels were responsible to produce the depolarizing component of slow wave, and this potassium efflux was responsible to produce the repolarizing component of slow wave. But if peak of the slow wave, the top of the slow wave touches the threshold, then threshold voltage sensitive calcium sodium channels open, and they allow a lot of calcium to trickle in. And when a lot of calcium come in, then this will move upward. As soon as it reaches to the, what is this? Membrane becomes fully depolarized. Is that right? And as soon as it reaches to the top, what really happens? Again, a lot of potassium goes out and it comes back. But as soon as it comes back, if still the slow wave is at threshold, it will again fire again. So what really happens, that if top of the slow wave is touching the, what is this? Threshold potential, then at the top of the threshold potential, action potentials are produced as spikes. So these are also called spike potentials. What are they called? Yes, spike potentials. These spike potentials which are produced, these are actually the action potentials. Slow waves are not action potentials. But remember, slow waves play a very major role in production of spike potential. How? If slow waves, top of the slow wave does not touch the threshold potential, then what will happen? Spike potential will not be produced. If top of the slow wave touches threshold potential, spike potential, multiple spike potential can be produced. And if it is, let's suppose smooth muscle is really very excitable and it is going significantly above the, what is this, spike potential, then multiple spikes will be produced. Is that right? So what is the basic principle? Number one, spike potential, right, can only be produced at the top of the slow waves, right, which is the real action potential, can be only produced when slow waves, top of the slow wave reaches threshold. Now how many action potentials will be produced? It depends on that how large is the slow wave and how long slow wave remain at threshold. Is that right? Because if slow wave does not reach its threshold, no spike potentials or action potentials. Slow wave just touches and come down, few spike potential. But if slow wave is really strong and remain above the, at or above the threshold potential, multiple spike potentials are produced. Now, because heavy amount of calcium influx is produced during the spike potential, that calcium act with the calmodulin system and that produces eventually smooth muscle contraction. And if I show smooth muscle contraction by this color, now here there will be no smooth muscle contraction, right? But here with a slight delay, there will be smooth muscle contraction. But in this case, because there are more spike potential, so intensity of contraction of smooth muscle will be stronger. So what I'm showing here I'm showing the slow waves. Here I'm showing the, what is this? Spike potential, the action potential. Here I'm showing that whenever spike potential appear, then the really smooth muscle contract. This is the tension in the smooth muscle. So tension in the smooth muscle goes up. But tension of produced in the smooth muscle is directly proportional to the heavy calcium influx during spike potentials, and that calcium influx is of course directly proportional to the frequency of the spike potentials. Here there are more spike potentials, right, and that produces larger or stronger smooth muscle contraction. Any question after this? There's no question. Right. Now, what we can say that electrical activity in the smooth muscles mainly depends on, what is this? Slow waves. Now, this also, the frequency of slow wave also determines the rhythmic contraction. There are two types of contractions in smooth muscles of gastrointestinal system. There is tonic contraction and phasic contraction. Tonic contraction means prolonged, persistent contractions of smooth muscle. And phasic contraction is intermittent contractions, or contraction followed by relaxation, again contraction followed by relaxation. Now these action potential, the spike potential, basically or primarily determine the what? Intermittent contractility. What is that? That is phasic, right? So phasic contraction depends on how many spike potentials are being produced at the top of slow waves. At the top of slow wave, right? Now another thing which is very important to understand is that not only amplitude of the slow wave can be changed, not only frequency of the action potential can be changed, in these smooth muscles you can also change the resting membrane potential. Is that right? Let me explain it. Already we have observed that if there is a slow wave is weak, there is no spike potential. If slow wave is stronger, there is a spike potential. And strength of the slow wave eventually determines the frequency of spike potential. And frequency of spike potential determines the intensity of smooth muscle contraction. Now we'll observe another thing. that this resting membrane potential, right, which is normally around minus 60, right, it can be persistently made more negative or it can be made less negative, right? There are some factors which move the resting membrane potential from minus 60 to minus 50 millivolt. And the other factors which can take the resting membrane potential from minus 60 to minus 70 millivolt. Now all those factors which take the resting membrane potential persistently near minus 50, they will excite the smooth muscle. Smooth muscle excitability will be increased because then most of the slow waves will go above the threshold potential and smooth muscle will be more stimulated, right? But if some factor take the resting membrane potential persistently more negative, right? So they are taking the resting membrane potential away from the threshold. Is that right? Then excitability of smooth muscle will be reduced. How? Let's suppose if excitability of resting membrane potential is moved from minus 60 to 50, what will happen? Then slow waves, right, they will more frequently remain above the threshold. So more spike potential will be there. But if, let's suppose that resting membrane potential is away from the threshold and it becomes more negative, we say it is all the time hyperpolarized, right? Then what will happen? Most of the slow waves will not reach to threshold. Threshold was here, right? And smooth muscle excitability is reduced. Now the question is that what are the factors which make the smooth muscles more excitable or take the resting membrane potential near to threshold, right? Number one is the stretch in the smooth muscle. If smooth muscles in the gastrointestinal system, they are stretched when GIT is distended. For example, there's big bolus of food here and this part of the GIT is stretched and smooth muscles are stretched. When smooth muscles are stretched, then these calcium channels like holes in an elastic membrane. When an elastic membrane has multiple holes and you stretch the elastic membranes, hole become wider. In the same way, when you stretch smooth muscle membrane, these channels become wider and more cations influx occur and stronger, and when the cation influx is more, right, what will happen? Resting membrane potential will remain persistently more near the threshold. Plus stronger slow waves will be produced. Is that right? So what are the factors which can take it upward? Right? Number one is, yes, muscle stretch. Number two is, yes, any condition will load the smooth muscle with the cations like acetylcholine. Right? There are muscarinic M3 receptors on smooth muscles of gastrointestinal system, muscarinic 3 receptors, and either acetylcholine like drug is there or vagus nerve, or parasympathetic, some other neurons present in gastrointestinal, you can say plexus, those neurons release acetylcholine. When acetylcholine stimulate the receptors, that lead to the calcium influx inside. And when calcium moves in, that takes the, resting membrane potential towards threshold. Is that right? So acetylcholine. And of course, third thing is parasympathetic nervous system stimulation. So these are the factors which can stimulate and some hormones even. There are some hormones which stimulate the electrical activity in the smooth muscle, the GIT. Actually, those hormones load the smooth muscles with the cations and take the resting membrane potential near the threshold. Is that right? So we can say resting membrane potential can be made less negatively polarized or slightly depolarized all the time, right, by stretching the smooth muscle or by acetylcholine or by stimulation of parasympathetic nervous system or by certain stimulatory hormones. Is that right? Then resting membrane potential, if it is taken away, it has been made more, negative, right? It means membrane is more hyperpolarized, right? Then smooth muscles become less excitable, right? Under what circumstances this can happen? This hyperpolarization of the membrane is seen when norepinephrine norepinephrine stimulate these smooth muscles, right? Because when norepinephrine stimulates these smooth muscles, right, that actually take the resting membrane potential away from threshold, and this become hyperpolarized, and natural excitability of the smooth muscle is reduced, right? Or, of course, by stimulation of sympathetic nervous system. Is that right? Do you have any question up to this? Or that, yeah, what is your question?
You're saying there's calcium leaky channels that keep on trickling calcium inside, and that maintains the frequency of the slow waves.
Yeah.
But I thought there were intracellular calcium stores also inside the cell.
Yeah.
The smooth endoplasmic reticulum.
Yeah. Actually, the hormones, you are right. Actually, the hormones can increase or decrease the excitability of smooth muscles by giving special signal to the calcium stores in the endoplasmic reticulum within the smooth muscle. If calcium is released from the endoplasmic reticulum, smooth muscle will become more excitable. If calcium is sequestered or stored in the endoplasmic reticulum, smooth muscles will become less excitable. Am I clear? Is there any question? No question? Right? So let's have a break. Now we are going to talk about the nervous control of gastrointestinal system. Right? First of all, I will discuss into detail the enteric nervous system, and then I will talk about that enteric nervous system activity is influenced by the autonomic nervous system as well. Now, what is enteric nervous system? Actually, it is also called the third division of autonomic nervous system. As you know, autonomic nervous system has one division called sympathetic part of the autonomic nervous system, then parasympathetic part of the autonomic nervous system. Then some authorities say the neurons which are present within the wall of gastrointestinal system, they should be considered the third part of the autonomic nervous system, or enteric nervous system. Enteric nervous system mainly consists of two neuronal plexes. One neuronal plexus is present in submucosa. Look, this is the mucosa, right? And this layer is submucosa. This is muscularis externa, circular layer, and longitudinal layer, and serosa. Now look, this is one layer of neurons which are present with their cell bodies and connections in the submucosa. This is called submucosal neuronal plexus. The other name for submucosal neuronal plexus is Meissner's plexus, right? Second neuronal plexus is present in muscularis externa, and second layer, or we also call it outer layer, is present in between the circular layer of smooth muscles and longitudinal layer of smooth muscles in muscularis externa. So these are the cell bodies and connections of outer plexus, which is also called myenteric plexus. Another name for this is Orback plexus. Orback plexus means this plexus is Orback plexus, or you can say outside plexus. This is an outer part, it is in the muscles. Now, what is the major function of submucosal? What is the major function of the submucosal plexus? Actually, submucosal plexus is mainly controlling the secretion of mucosal and submucosal glands, plus submucosal plexus also control the blood flow through the submucosa and lamina propria, and that also determines the absorption. So it regulates secretion, absorption, blood flow in this area, plus it's also, submucosal plexus also control and has influence on neuroendocrine cells which are present in this area, right? An important thing is that submucosal plexus usually control locally with local areas and small areas within the gastrointestinal system, right? But if we talk about the outer plexus, what is outer plexus? The other name for outer plexus is myenteric plexus. Another still name is Orback plexus, which is present between which two layers of muscularis externa, circular layer of muscles and longitudinal layer of muscles. Now this plexus of neurons is mainly concerned with the regulation of motility of gastrointestinal system. Now let me tell you exactly how it works. Enteric nervous system. Remember, enteric nervous system can work independent of outside neuronal control. if you disconnect all the sympathetic and parasympathetic fibers coming to gastrointestinal system, still these neuronal components of gastrointestinal system, that is submucosal plexus and myenteric plexus, still can work independent of the autonomic fibers coming to the gastrointestinal system. That is why sometimes the gastrointestinal, you can say, these neuronal plexus are also called mini brain. Is that right? They can be influenced by autonomic supply, sympathetic and parasympathetic system, but they have their own intrinsic activity as well. Is that right? Now, how the myenteric plexus work? Let me give you an example. We'll talk about how this nervous system works to create peristalsis. Let us suppose that, okay, you have eaten a chicken piece, right? Let's suppose there's a bolus here, amount of food here. Now this area will be stretched, right? Now this stretching of this area, right, how it will influence? These are the sensory neurons, I think. These black neurons are the sensory neurons. So let me tell you. Now what really happens when food is stretching your gastrointestinal system? Is that right? In the lumen, if there is some stimulus, let us suppose stimulus is a large mass, it will stretch. When it will stretch, it will stimulate, what is this? A sensory neuron which is present in the muscle layer, and when this part of the gastrointestinal system is stretched, these muscles are also stretched, and these neurons are also stretched. And this neuron is stimulated, right? And this will stimulate, what is this plexus? Outer plexus or myenteric plexus, right? So one way to stimulate the myenteric plexus is by stretching a part of gastrointestinal system. Secondly, it can also be stimulated by giving some signal to the mucosa. Let's suppose there's chemical signal to the mucosa, or there's some irritation to the mucosa, right? Again, there are other sensory neurons. They will take this information and they will stimulate the, what is this neurons? Part of the myenteric plexus. Now let's see that we say that when there's a bolus, this should produce peristalsis. The law of peristalsis is that to the oral side of the stimulus, gastrointestinal system should contract as a ring, and on the anal side or aboral side, the smooth muscles should relax. Now we have to see how this system works to produce a contraction just, yes, oral side of the stimulus, and how it produces receptive relaxation on the distal side of or anal side of the stimulus. Now we want that if peristalsis should produce, these muscles should contract and these muscles should relax, and this wave of contraction should pull the content of GIT towards the anal side. How exactly it happen? Now these sensory neurons from here as well as stretch neurons from here, they stimulate these fibers. Now I have shown, and look it carefully, that in myenteric plexus I have shown two interconnected chains of neuron. These are the interconnected chains of neurons, right? And they are going to the oral side. We also call it ascending network. And these are the network of interconnected neurons which are mainly moving to the anal side or aboral side. These are also called descending network of neuron. So we can say in myenteric plexus there's ascending, right, network of neurons, and there's descending network of neuron. And what we want? On ascending side, we want contraction. And on descending side, we want relaxation. It means on ascending side, we want stimulation. And on descending side, we want inhibition. How it really happens? When sensory neurons from mucosal side and from other areas, they stimulate the ascending neuronal network as well as descending, what will happen? First look at ascending. When ascending neurons are stimulated at this point, right, then what will happen? That these neurons will fire in this direction, right? And they will produce actually stimulatory neurotransmitters. These neurons produce acetylcholine and substance P. acetylcholine and substance P, these are stimulatory neurotransmitter and they are produced by the ascending neurons. Now, when substance P is produced, what it will do? Substance P and acetylcholine, it will stimulate the neurons above the stimulus, or stimulate the neurons proximal to the stimulus, or stimulate the neurons in the ascending area, or stimulate the neurons which are on the oral side. When these neurons are stimulated, they will release their neurotransmitters on these circular, especially they will stimulate, what is this? Circular side. And circular smooth muscles will contract on this side. Right? So what will happen? The constriction will be developed right on the back of, what is this, chicken? Right? So when this will contract, right? So at the same time, these sensory black neurons have also stimulated descending neurons. Now, when the descending neurons, they are stimulated by mucosal stimulus as well as stretch stimulus. When they are stimulated, these descending neurons release neurotransmitters which are inhibitory. What are these neurotransmitters? These are inhibitory. These neurotransmitters which are released by these descending neurons, they are nitric oxide and they are, yes, what is it? VIP, vasoactive intestinal peptide. Vasoactive intestinal peptide, right? So both of them are inhibitory. So what happens that when this neuron is stimulated, it will start producing what kind of neurotransmitters mainly on which layer? Longitudinal, and some inhibitory neurotransmitter on the circular layer. So what happens, then circular smooth muscles will start getting relaxed, and when circular smooth muscle gets started relaxed, right, there will be receptive relaxation, right? And so just aboral to the stimulus, circular smooth muscle will relax, and on oral side of the stimulus, circular smooth muscle will contract. Is that right? And this piece of bolus will be pushed forward. Then what will happen? The new sensory neurons will stimulate the descending and ascending neurons, and ascending neurons will produce stimulatory effect at new site stimulatory effect, and descending neurons will produce inhibitory action on more distance to the anal side and receptive relaxation. And again, peristalsis will move, constriction ring will move forward. Is it clear or not? So this is one way how the enteric nervous system works. Again, let me repeat it. Enteric nervous system has a lot of neurons. The number of neurons is almost about 100 million. You're a millionaire. As far as these neurons are concerned here in the enteric nervous system. These neurons are almost equal to the number of neurons present in spinal cord. So it means a very important nervous system. So you must understand. Enteric nervous system has two parts. Submucosal part and what is this? Enteric part. Submucosal part is mainly concerned with secretions from mucosa. It is concerned with regulation of secretions, absorption, and blood flow in this area. Plus, submucosal neuron also control the muscles which are muscularis mucosae, a thin layer, which produces infolding in the mucosa. But the real motility controlling neurons are myenteric. And myenteric neuronal plexus is having interconnected neurons which are ascending network and interconnected neurons which are descending. And ascending are stimulatory and produce acetylcholine and substance P. And descending are inhibitory and they produce nitric oxide and vasoactive intestinal peptide. Is that right? Now, what you need to remember is that whenever these myenteric plexus is stimulated, ascending, when they get stimulated, they stimulate the circular layer just above the stimulus, on the oral side of the stimulus. And descending neuronal system, when it is stimulated, it inhibits the circular neurons just aboral to the stimulus. Am I clear? Any question up to this? Now, if someone asks you what are the difference, again, submucosal is concerned with secretion, absorption and and neuroendocrine secretions of these cells and blood flow in the mucosa and submucosal area. And what is this? My enteric is mainly concerned with the motility of gastrointestinal system like peristalsis and mixing movements. Then we come to that enteric nervous system is also influenced by the sympathetic and parasympathetic nervous system. Now, parasympathetic nervous system is supplying to the enteric nervous system of GIT through cranial vagal outflow mainly, and from the sacral outflow from S2, 3 and 4 through the pelvic nerves. From the vagus nerve, preganglionic fibers come up to the gastrointestinal system, and these preganglionic fibers directly innervate the neurons in enteric plexus as well as neurons in submucosal plexus. Is that right? And actually, you can also think that these neurons, right, or cell bodies of neurons in myenteric plexus and submucosal plexus can be considered post-ganglionic neurons of parasympathetic nervous system, right? But sympathetic nervous system, preganglionic fibers terminate in prevertebral ganglion, like celiac ganglion or superior mesenteric ganglion or inferior mesenteric ganglion or hypogastric ganglion, right? So from there, post-ganglionic sympathetic fibers come and they influence the, what is this, myenteric plexus and submucosal plexus. Now the very important role which you have to understand is what is the effect of parasympathetic stimulation and sympathetic stimulation on the gastrointestinal system. Let's discuss it conceptually. You know, sympathetic nervous system is stimulated when we are under stress. And do you think when we are under stress, we want our gastrointestinal system to work? No. Because when you are under stress, fight or flight response, when you are going to fight, you don't want that movement to gastrointestinal peristalsis or secretions. You want to take blood, you want to reduce, when you are under stress and sympathetic nervous system is firing, you want to reduce the work in GIT. You can digest the chicken piece later. First you have to fight or flight. Is that right? So sympathetic nervous system is primarily inhibitor to the gastrointestinal system through the effect on the, what is this, enteric nervous system. Also sympathetic nervous system directly control the vasculature in gastrointestinal system. And parasympathetic system is considered rest and digest system. Actually, parasympathetic stimulation enhances the activity in gastrointestinal system. Parasympathetic system enhances all the activity of GIT. It increases peristalsis, it increases mixing movement. Parasympathetic nervous system also increases the glandular secretions in gastrointestinal system. Now look, if you have taken good amount of food and you don't have much stress, your parasympathetic system is working well, and parasympathetic, for example, vagal fibers, they can come and stimulate the myenteric neurons, and that will lead to more peristalsis and mixing movement. And parasympathetic fibers will come to the submucosal area and stimulate more secretions, right? And they will also stimulate more absorption. So they help in digestion. Is that right? Because when peristalsis and mixing movement are more, right, then food is not only mixed with the secretions, but also it is propelled forward. And when, of course, secretions are more, digestion is more. And when food is moving forward, it is also absorbed better. Is that right? With mixing movement and peristalsis. So parasympathetic nervous system increases the peristalsis. It increases the, yes, parasympathetic nervous system increases the frequency of peristalsis, even intensity of peristalsis. Is that right? And also increases secretions and eventually absorption also. But when we talk about sympathetic nervous system, sympathetic neurons which secrete norepinephrine, they basically inhibit the orbic plexus as well as myenteric plexus. Then of course you can understand when these two plexes are inhibited, when myenteric plexus is inhibited, mixing movement and propulsive movement, peristalsis are inhibited. And when orbic plexus is inhibited, it's very understandable that will reduce the secretions in gastrointestinal system. Plus, sympathetic fibers directly also act on the blood vessels in gastrointestinal system and constrict the blood vessel. It's very important. Sympathetic neurons directly innervate the subplanchnic blood vessels and blood vessels of gastrointestinal system and produce strong vasoconstriction, so that blood should be diverted to other organs where we need more blood during stressful situations like skeletal muscles. Maybe you are planning to fight or flight. Is that right? Do you have any question up to this? That what is enteric nervous system? Can you answer and can you tell there are two divisions, right? Enteric and submucosal. You know submucosal work in local areas and concerned with secretion, absorption and local enfolding of muscularis mucosae and blood flow in the mucosa. And what is this? Enteric is mainly concerned with the motility, and sympathetic mainly inhibit the gastrointestinal activity and reduces blood flow to GIT. Parasympathetic increases the motility of the GIT as well as increases the secretory activity of gastrointestinal related glands. Is that right? Any question here? There's no question. Okay, that's good. Now I will talk about the certain reflexes which are related with gastrointestinal system, right? But again, don't forget, in the gastrointestinal system there are sensory neurons also. There are some short sensory neurons which have their cell bodies within the wall of GIT, and they just take the sensation from the mucosal side to the enteric plexus, and there are other local sensory neurons which take stretch sensation to the plexus, enteric plexus. But there are other sensory neurons which take sensation along with the vagus. You know, 75% of the fibers in vagus nerve are sensory. So these many sensory neurons from gastrointestinal system, they take sensation from mucosa and also stretch sensation and distension sensations, and they take all the way along with the vagus nerve sensation to the medulla. Is that right? And then there are also more sensory neurons which will take sensations from gastrointestinal system to the spinal cord. And something which usually students forget, that there are also sensory neurons from gastrointestinal system which take sensations up to the sympathetic ganglion. So how many types of sensory fibers are present in gastrointestinal system? Number one, sensory fiber for local reflexes, then sensory fibers up to the sympathetic ganglion mediated reflexes, then sensory fibers which are taking sensations up to the central nervous system, either through the vagus to the medulla or through other connections to the spinal cord for the reflexes which involve the central nervous system, right? Let me explain these reflexes very briefly. Now, there are basically three types of reflexes related with the neuronal reflexes related with the gastrointestinal system, right? And of course, like reflex, let me draw a diagram and explain it well. Now, let's suppose that here is your gastrointestinal system. This is a very simple diagram, right? Now listen. There are some reflexes, some neurons, sensory neurons which take information from one part of gastrointestinal system to another part of gastrointestinal system, right? So these are for the local reflexes. For example, if there's irritation here, more secretions will come. Or if there's distension here, peristalsis will start over here. These are intra-gut reflexes. What are these reflexes? These are intra-gut reflexes. These are for the secretions and mixing contractions and peristaltic contraction. Then, as I told you that central nervous system and sympathetic ganglion also play a role. Let's suppose this is preganglionic sympathetic fibers and this is postganglionic sympathetic fiber going to gastrointestinal system. Now, some sensory fibers can go from one part of gastrointestinal system up to the ganglia. Is that right? And then response come. For example, fibers from here, sensory fibers from the stomach, can, when stomach become full, they can take sensations to a ganglion, and from that ganglion, fibers get connected to other part of gastrointestinal system, right? Now these are the reflexes which involve the ganglion, right? And these reflexes, right, these are called reflexes from the gut to the prevertebral sympathetic ganglion and back to the GIT, right? Classical example of this is gastrocolic reflex, that when you eat and your stomach is full, this reflex increases the motility in the colon. What is this called? Gastrocolic reflex. Then there is another reflex in which if small intestine or large intestine is overloaded, right, overloaded with the luminal content, it should inhibit stomach. And this reflex, right, sensory fibers go up to the ganglion and then motor fiber inhibit, what is this? Stomach. So this is called enterogastric reflex, right? So first was content of stomach lead to inhibition of stimulate colon. That was which reflex? Gastrocolic. Second reflex I told you, this was stimulatory. That when the contents of small intestine and large intestine are overloaded, luminal content, they will inhibit the stomach. So that is called enterogastric reflex. Gastric reflex, and that is inhibitory. Reduce the secretions and reduce the motility of stomach. Then there is another reflex which is mediated through the ganglion, in which there is basically coloileal, that if colon is overloaded with the content, it will inhibit the ileum and stimulate the contraction of ileocecal valve, so that contents should not move very rapidly to the overloaded colon. And that is called which reflex? Yes, coloileal. Some people call it colonoileal. So these are three types of reflexes which involve the sympathetic ganglion. That sensory fibers go up to the sympathetic ganglion, and from there the motor fibers come back. Right? Then another type of reflexes are, I told you there are basically three types of reflexes. Intragut reflexes, secondly in reflexes involving the sympathetic ganglion, third type of reflexes in which sensory fibers go up to the central nervous system. For example, some sensory fibers from the stomach can go up to the, along with the vagus, up to the medulla, right? And then vagal response come back to the stomach, right? This is called vagal reflexes. Is that right? Which control the motility and the secretions in the stomach, right? Because sensory limb is also going through the vagus and motor limb is also coming through the vagal. So such reflexes in which sensory fibers go through the vagus to the medulla and then motor fibers come back through the vagus, what we call it? Vagal reflexes. Is that right? Then another classical example of reflex in which central nervous system is involved is when sensory fibers from colon and rectum, they go to the spinal cord, and then from spinal cord motor fibers come back and there is defecation reflex. That when colon and what is this part? Last part of the colon and sigmoid, that is overloaded with the fecal matter, overstretched, and that can stimulate a defecation reflex, and motor fibers are coming back through the spinal cord. Is that right? So how many types of reflexes? Intragut reflexes, which are local reflexes, which produce secretions, right? And they regulate the secretion and absorption. Is that right? Then there are ganglionic reflexes like gastrocolic, enterocolic, and enterogastric and coloileal. And then there are reflexes involving the central nervous system like vagal reflexes or defecation reflex. Class dismiss. Today we are going to talk about the functional type of movements in gastrointestinal system. Basic movements or functional type of movement. Functional type of movements, right, in gastrointestinal system. The major movement in gastrointestinal system, they are divided into two parts. Is that right? What are the two main types of the movement? Yes, who will tell me? What are the two main types of movement in gastrointestinal system? there are only two types. Look, number one, there should be propulsive movement. We should propel the food or the chyme forward. Look, for the proper processing of the food, what is required? For proper processing of the food is, number one, you should be able to ingest it. It's of no benefit on the plate, right? And after ingestion and swallowing, when it goes into what is the stomach and small gut and large gut, what should happen there? Number one thing, food should stay in different areas of gastrointestinal system for appropriate duration. It should not understay or it should not overstay, because different areas in the GIT process the food in a different way. Is that right? So for this purpose, number one, there should be propulsive movement. We should propel the contents in the GIT forward from oral side to the anal side. Is that right? We have to use the propulsive movement. Secondly, on the way, not only we need to propel the food, we have to mix the contents of food with the gastrointestinal secretions, so that secretions and contents of food get mixed, so that secretions can work on the gastrointestinal food stuff and digest it, right? For that purpose, we need to do mixing, right? So second type of the movement should be mixing. So there are basically two types of movements. There are propulsive movement and there are mixing movement. Is that right? When we talk about the propulsive movement, remember, propulsive movement are an inherent feature of all those tubes in our body, where smooth muscles are connected with each other through the gap junctions. Multiple smooth muscles are connected with each other through the gap junctions. Their propulsive movement can be produced. For example, not only gastrointestinal system, even in ducts of the glands, they also have propulsive movement. Then propulsive movement are also seen in ureters. Is that right? Now, so this is propulsive movement is an inherent tendency of those tubes in our body which have smooth muscles in their wall, and those smooth muscles are connected with each other with the gap junctions. Is that right? When a lot of smooth muscles are connected with the gap junction, what do we call this type of smooth muscle? Unitary smooth muscle. They act as a unit. Is that right? There may be 10,000 smooth muscles, but they are connected with each other through the gap junctions, so they are acting like a network or syncytium. So we call them unitary smooth muscle. Now GIT has unitary smooth muscle, and propulsive movements is a feature of unitary smooth muscles. Now, what is the purpose of propulsive movement in the gastrointestinal system? It's very clear that they are moving the contents of the lumen, right, from one point to the next point, usually from oral side to the anal side. Is that right? Now, propulsive movements are also called peristalses. Peristalses. Classically, we can explain peristalses as, right, let me tell you, suppose this is the gastrointestinal system, right? Now, what is basic concept of peristalsis? Peristalsis mean that, let's suppose there is a bolus here, or there is accumulation of chyme here. Now what will happen this need to be propelled in this direction to the anal side. If it need to be propelled, how the system start? It's very simple. Number one, this bolus, right, will distend the wall of gastrointestinal system. And when it will distend the wall of gastrointestinal system, that will activate the stretch receptors. Let me enlarge this part of gut here, right? This part. And now there are stretch reflex into this area. When they are stretched, when stretch neurons are stimulated, they will stimulate the enteric plexus. As I told you in the previous video, that enteric plexus has two layers. It has ascending neurons and descending neurons. And ascending neurons are stimulatory and descending neurons are inhibitory. That's right. Now, enteric plexus can be stimulated, number one, by the stretch in the wall. Number two, it can also be stimulated by special chemical substances like amino acids or like fats, or there are different substances within the food which can stimulate some chemoreceptors here in the mucosa. So, right, in the same way, if there's irritation to the mucosa, that can also stimulate certain sensory neurons here. So what happens? That either stimulation is coming from mucosal irritation, or mucosal stimulation by certain type of chemoreceptors, or stimulation is coming from the stretch receptors, right? And this will stimulate the enteric plexus. Now what really happens? That when enteric plexus is stimulated, right, then these neurons which are ascending pathway, they release stimulatory neurotransmitters like acetylcholine and substance P. And these neurons which are descending, right, they release inhibitory neurotransmitters. What are those? VIP, vasoactive intestinal peptide, and nitric oxide. So it's very natural that just in the part segment of the gastrointestinal system, just on the oral side of the stimulus, acetylcholine and substance P will be released, and to the distal side, let's take the oral side as proximal side, and let's in my lecture we take what is this anal side as distal side. So proximal to the bolus, stimulatory neurotransmitters are released by enteric plexus, and distal to the bolus, inhibitory neurotransmitters like VIP and nitric oxide are released. What happens? Smooth muscle just proximal to the stimulus will constrict and produce a constriction ring. But at the same time, muscles in the muscularis externa distal to the stimulus will get relaxed. Now it's very natural when you produce a constriction ring on the proximal side and relax the wall of the gut distal to the stimulus, what will happen? It will build the pressure. There is more pressure on the contents of the lumen on the oral side and less pressure on the anal side, so it will move forward. When it will come to the new place, let's suppose, then it has shifted to this place now. When it has gone to the new place, now at this new place, stimulatory neurotransmitters will be released. And just in front of that, there will be inhibitory neurotransmitters will be released. By which plexus? Enteric plexus, right? So what will happen? It will slip forward. And then this process will repeat again and again. So due to this mechanism, we can say that a propulsive movement will be produced, a movement which can propel the contents of the GIT from oral side to the anal side. This is clear? Right. Now, another thing which is important, that this ring, once it develops, how much it will move, it depends on the general excitability of the gastrointestinal system at that very moment. For example, if there is strong parasympathetic stimulation, right, then this peristalsis will move for longer distance. But if there is extrinsic nerves, like parasympathetic nerves, right, they are not stimulated and not stimulating the enteric plexus enough, then the peristalsis will move for smaller distance. Am I clear? So even though this movement peristalsis is produced within the enteric reflex, but it will be a weak peristalsis, a strong peristalsis. It depends on many factors, including the extrinsic neuronal control like parasympathetic or sympathetic stimulation. Here I want to know that everyone knows parasympathetic system stimulates the gastrointestinal activity. What about sympathetic system? That inhibits. Is that right? That the sympathetic nervous system inhibits the gastrointestinal activities. Anyway, so this is something about propulsive movement, right? This is how they are produced. Then a part of the GIT, there is a constriction ring produced, and that constriction ring move from oral side to the aboral side, right? Taking the contents, so luminal content pushing towards the anal side. Then there are other movements which are called mixing movement. Here I want to make it clear. Propulsive movement can do some degree of mixing also, right? Of course, when contents of the lumen are pushed forward, on the way some secretions are added and some mixing will be done, right? Especially in the stomach, so a weak propulsive movement do the mixing, which I will explain later. Now, mixing movement. Let us suppose right now this area is not undergoing propulsive movement, rather it is busy in mixing. Is that right? Remember, purpose of propulsive movement is different, purpose of mixing movement is different. Purpose of mixing movement was to mix the secretions of gastrointestinal system with the food content, right? So that there should be good digestion. Plus, repeatedly mix the contents of the lumen and bring different part of the luminal content with the mucosal wall for better absorption. Is that right? But purpose of propulsive movement is the food has been, let's suppose, processed in the stomach properly. Only then it should move to the duodenum, right? If it has been properly processed in the duodenum and jejunum and ileum, only then it should go to the colon and so forth. Is that right? Now, let me tell you in a very simple way that what can be the example of mixing movement. Mixing movement, let me show you one mixing movement here. Let's suppose this is segment of GI tract. Now what happens? In mixing movement, localized constriction ring develop at multiple places which do not move, because if they start moving too much, then that is peristalsis and that becomes propulsive. But if simultaneously multiple constriction ring appear, for example, smooth muscle here contract, here contract, here contract and here contract. So what will happen? That gastrointestinal system, it will become like this here, and again it will become. So what is happening? That contents of the lumen will be compressed at multiple places. Now here what I am showing, that luminal content, whatever are there, now they are chopped down into multiple pieces, right? So multiple circular smooth muscles constrict at multiple points simultaneously, and luminal contents are divided into like chain of sausages, right? It remains just few seconds like this, and then these points relax and some other points contract. Let's suppose all of them suddenly relax and now these points constrict. If these points constrict, now what will happen? These are different points now which are constricting. Constriction point is here, and what is happening? This will become constricted here, and again it is opened here and constricted here, then here and here. Now you can understand what has happened. That same, let's suppose if this was the one area, now due to new constriction point which appeared in between the previous 2 points, it chops it off to this side and this side. So, you got it? Now this point, when their constriction appear at this point, this goes partly there and partly here. If this is constriction point appear here, so and so forth. So what is happening? Again, in mixing movement, when repeatedly multiple circular constriction appear simultaneously within the GIT and divide the GIT into small segments, then we call such movement in small intestine are called segmental movements, right? And these segmental movements, special thing is that at different times these segmental constrictions appear simultaneously but at different points. In this way, they chop the contents of the lumen of the gastrointestinal system here and then there. And repeatedly by this process, they mix these contents of the lumen with the different secretions, plus they bring different components of the food or luminal content in contact with the mucosa for absorption purpose. So what are the basic 2 movements? The propulsive movements, classically peristalsis, and the mixing movement like segmentation movement in small intestine. Is that right? Do you have any question? Now we come to the role of myenteric plexus in these movements. In previous video we have discussed in detail how myenteric plexus work. But one thing is very important. If due to any reason my enteric plexus is not functional, what could be the reason my enteric plexus is not functional? If it is absent, congenital absence of my enteric plexus. If in some segment of the gastrointestinal system my enteric plexus is absent, is that right? For example, neural crest cell did not migrate to that segment of the gastrointestinal system, and there the ganglia of the enteric plexus are not there. You can say a ganglionic segment is there. Do you think that will produce mixing movement significantly, or will produce some significant peristaltic movement? No. So it means my enteric plexus is important. Number two, if you give atropine to a person, and if a higher dose of atropine is given, atropine block which receptors? Muscarinic receptors for acetylcholine, right? And acetylcholine was one of the stimulatory neurotransmitters in gastrointestinal system. But if you give the person atropine, and atropine block most of the muscarinic receptors for acetylcholine, then acetylcholine fail to work on gastrointestinal smooth muscle, do you think you can get much mixing movements, or you can get much propulsive movement? No. So you have to remember it, for GIT movement, for significant or effective gastrointestinal movement, I'm talking about from esophagus down up to anus, we have to have what? My enteric plexus. Is that right? The next thing, there is a very simple concept called the law of gut. What is law of gut? There is some high court there or Supreme Court? Law of gut. Yes, what is law of gut? It's a very simple concept. I don't know why they try to make it a law. The thing is that our gastrointestinal system has enteric plexus polarized in such a way, or arranged in such a way, that whenever you distend any part or stimulate any part of gastrointestinal system, peristalsis which is produced by the enteric reflex, as I told you, that whenever there is stimulus or stretch, right, what happens? Peristalsis is produced if my enteric system is intact, right? So whenever you produce a stimulus in any point in the gastrointestinal system, right, if enteric reflex is intact, the peristalsis which will be produced will move more to anal side, and it will die out to the oral side. You are understanding? Thank God for this thing. Otherwise, you know what may happen? One day you forget you have to move the things in which direction. Yes. So thank God it is not in your will, it's automatically happening. That nature has designed the enteric plexus in such a way that whenever there is significant stimulus to enteric plexus and peristalsis is generated, it dies out on the oral side, and it effectively moves, the constriction ring effectively moves towards the anal side. This is called law of the gut. That this is the law of the gut, that when enteric reflex is intact and stimulus for peristalsis is given, peristalsis will more effectively move from oral side to the aboral side or anal side. Am I clear? I told you this topic is very easy. It should be banned to teach it. Now we have talked about okay, after this basic concept about the movement, now let's start step by step. What are the special movement related with the ingestion of food, like mastication and swallowing? What are the special movements in stomach? What are the special movement of small intestine? And then of course in the end, what are the special movement in large intestine? And then we'll study what is defecation reflex. Is that right? Now, part by part, we'll discuss the motility in the different part of the gastrointestinal system. Let's start with the very simple ingestion. Now we are going to discuss in this order. First we'll talk about ingestion of food. The first part of ingestion is what? Chewing. Yes, chewing. After that chewing, then we are going to talk about, yes, swallowing. And after that we are going to talk about motor functions of stomach. Motor functions of stomach. Now, first of all, chewing. Everyone knows what is chewing, that you put some eatable substance in your mouth, and then you start chewing it, grinding it, cutting it, right? Our teeth are very appropriately designed. You know that frontal teeth, incisors are very good for cutting purpose, and teeth in the back, they're very good for grinding purpose. Is that right? But this chewing, this is reflex activity or voluntary activity? It is both. No, it is both. Chewing can be done voluntarily. Even sometimes you like to chew the things which are not eatable with your own choice, and you know with what pressure to apply and how much to apply. Is that right? Chewing can be voluntary. At the same time, you may put chewing gum in your mouth and you start moving your jaw, and then you start doing some other work and you forget that you are moving your jaw. Then chewing reflex has taken its work. Secondly, thank God, reflex component is there, because when you are eating, do you think with every chewing step you are memorizing where to move the tongue and how to move the teeth? This is almost automatically occurring. So chewing is voluntary as well as reflex activity. Thank God reflex activity is there. Sometimes we are doing some work, and while we are chewing something, it's automatically going on. So we should know about the chewing reflex. Who will tell me about chewing reflex? Yes, anyone? It's very simple. Look, let me tell you. Of course, when you put some, let's suppose you have taken some piece of food in your mouth. Is that right? Okay, before I really go for chewing, what is the difference in hunger and appetite? Don't tell me the spelling difference. Hunger and appetite.
Is hunger like a sign of that you need food, and appetite is kind of like, I don't know, like a desire.
Okay, let me tell you before he complicates the issue. Hunger is, listen. Hunger is intrinsic desire to take food. But appetite is when you preferentially seek for a specific type of food. Is that right? If you say I have to eat KFC and I will miss all other things, that is appetite for KFC. Is that right? But hungry means you just want to eat. And what you want to eat and you are seeking a specific type of food, then you are having appetite. Is that right? So let's go back to the chewing. Chewing reflex, yes. It's very easy. You put something in your mouth, right? And then what happens? You press the food. Is that right? Pressure in oral cavity goes up. That inhibits the muscles of mastication. What are the muscles of mastication? Muscles of mastication, main muscles of mastication I will talk about, which are the muscles which close the mouth. Temporalis, masseter, and medial pterygoids. And which are the main muscles which open the mouth? Lateral pterygoids, right? Okay, I will not go into detail how lateral pterygoid open the mouth. But what really happens when you put something in your mouth, after that, when initially you voluntarily compress, is that right? In the oral cavity, pressure goes up. And then receptors in oral cavity, they give signal to the brainstem, and that inhibits the muscles of mastication. When muscles of mastication are inhibited, jaw will drop. As soon as jaw drops, it elicits the stretch reflex. You know, as on the muscles you can produce stretch reflex. As the jaw drops, it stretches those muscles of masseter and temporalis and medial pterygoid. When that stretch reflex is elicited, muscles will contract. There is rebound contraction. When they will contract, you will chew. But as soon as pressure will go up in oral cavity, what happens? You again inhibit the muscles of mastication. Again jaw drop. As soon as it drop, it again produces what? Stretch reflex. So this cycle is repeated again and again, and you keep on chewing, depending upon how long you want to do it. Is that right? So this was your chewing reflex. And once you have chewed what is the advantages of chewing or mastication? Yeah, of course, we break down the food particles. I know you don't chew the water, right? You break down the food particles. There are 2-3 advantages. One is very simple, that you are mixing the saliva with that, right? During mastication, saliva get mixed, right? And salivary enzymes can also, amylase can start working on carbohydrate. Secondly, when saliva gets mixed, the bolus gets lubricated. Is that right? Thirdly, pieces of the food become very small. Now, when the bolus become lubricated properly and food pieces become very small, another advantage is then food will go down. There is less chance of excoriation or damage to the mucosa. If you take big bolus and without proper grinding and chewing, you swallow, you may damage your esophagus mucosa. Is that right? Thirdly, when you are grinding the food particles and making them smaller, you are increasing the surface area. And you know, enzymes in our gastrointestinal system work on the surface of the food particles. And if you are increasing the surface area, enzymatic digestion becomes accelerated. Am I clear? So anyway, you have done the chewing. The next step is swallowing. Right? To explain the swallowing, I should make a diagram here. Here you are. I think your nose should be smaller, or it's okay? Right? Okay. Now, oral cavity, there's tongue here. What is it here? Tonsil on that side. And what is this? Pharynx. Here is, what is here? Larynx. And what is this? Epiglottis. And here is trachea. Right? Now, the important point which you need to learn here is that, especially in the pharynx, this area, which is this part of the pharynx, work for respiratory passage as well as it can work for food passage. So we have to be very careful at this stage that food should not go to the respiratory passage, it should go to the food passage. Now, swallowing. What is swallowing? Swallowing, the process of taking the bolus of the food from posterior part of the oral cavity to the stomach. Is that right? Of course, don't forget, there is a stomach. I think I made it too much. Okay. Now, this is your esophagus. Now, in the swallowing process, how it starts? You have done enough chewing, and if you feel the food is ready for swallowing, you will take the bolus backward. Where you will take it? With the tongue, you will push the food bolus backward. And this is the early stage of the swallowing. We also call it oral stage of swallowing. There is oral stage of swallowing, there is pharyngeal stage of swallowing, and there is esophageal stage of swallowing. This initial stage of swallowing is voluntary, right? In this, what happens that voluntarily, once you think that you have chewed the food enough and bolus is ready to be swallowed, what do? You move your tongue upward. You move your tongue upward. When you move your tongue upward, what really happens? You press the food bolus against the palate, right, and push it backward. When it moves backward, right, on the opening of the oropharynx, there are a lot of sensory receptors for the swallowing purpose, right? The mucosa here, right, what is this area? Hard palate at the top. On the side there are tonsillar pillars and tonsillar mucosa, and then lingual mucosa. Is that right? Now all this mucosa here is very, very sensitive. As soon as you take the tongue upward, push the bolus backward, and bolus come in touch with this area and their sensory system, that triggers the swallowing reflex. So where the swallowing reflex is triggered? When you move the tongue upward, move the bolus backward, right, while pressing it against the hard palate, and when it has gone enough posteriorly, right, where especially in between the two tonsillar pillars, there are tonsillar pillars on both sides. They are the richest areas in sensory receptors for swallowing purpose, right? Now, as soon as you will stimulate this area, what will happen? That sensory system will take sensation to the, central nervous system. Right from here, which nerves will take information? Yes, 5th and 9th. Is that right? From anterior to 3rd, this touch pain temperature from the 5th, plus here also 5th nerve, trigeminal sensory part, is that right? And 9th nerve, glossopharyngeal sensory part, posterior part of the tongue. So 5th nerve and 9th nerve, they give what? Sensory input to the central nervous system, to the swallowing reflex. right? Swallowing reflex center. Let's suppose this is swallowing reflex center, which is very near the respiratory center in medulla, and swallowing reflex center is present in reticular formation and very near to the tractus solitarius nucleus, right? Now, there's swallowing reflex in the brainstem and upper medulla and lower pons. Now, as soon as food bolus reaches at this point, sensory system is activated, right, about the presence of the food. From here, now, what happens? Motor nerves will come out. Now, motor component will come out through which? They will come out through fifth. You know, fifth has a motor component also for muscles of mastication, plus through ninth glossopharyngeus, the motor component for stylopharyngeus, and from 10th nerve. You know, 10th nerve has taking a lot of fiber from nucleus ambiguous. You know that? Branchiomotor fibers, right? These fibers from the 10th, they are also going to, motor fiber from the vagus nerve, they will also play a role in swallowing reflex. Now, so sensations are coming out through 5th, 9th, 10th, and if tongue is also moving, then it is coming from which nerve? Tongue is moved by 12th these days, right? So, there's sensory input, central coordination, and then motor output come. Now, all these motor nerves, right, they are going to contract the muscles in this area in such a way that bolus will be transferred from here to the esophagus. But what are the changes here? Number one, the very first thing which we want is, look, what is this? What is this hanging here? Yes, soft palate, right? Well, of course, you realize the part of soft palate, right? Now, first thing, now if bolus come here, let's suppose swallowing reflex is not working. Now, Mr. Bolus is here. What are the choices for it? It can go to the nose, it can go to the respiratory system, it can go to the system. And unfortunately, normally, upper esophageal sphincter is closed or open? Yes, normally, upper esophageal sphincter is closed or open? It is closed. Why normally upper esophageal sphincter is closed? Yeah? Pardon? Okay, no, I'm saying when you are not eating, upper esophageal sphincter is closed, significant tone. Why? Yes? Yes, so that air does not enter inappropriately in gastrointestinal system. Is that right? The air which comes out through anus is not coming through mouth. That is produced somewhere distally. Is that right? So, actually upper esophageal sphincter is normally closed. And lower esophageal sphincter is also normally closed in between the swallows. Why? Upper prevent the entry of the air here and lower prevent the reflux of acid from the stomach. Is that right? Now, look at this. If bolus moves here, now what are the chances? It can go to the nasal side. Is it appropriate way to get it there? No. So, in pharyngeal stage, as soon as it will move backward, now it is going into pharyngeal system. At the pharyngeal stage of swallowing, number one, soft palate will move from down, it will move upward. Why it has moved in that direction? To prevent the movement of the bolus towards the nasal cavity or nasopharynx. Is that right? Secondly, most important thing is that when the food bolus is moving, we don't want any mistake that food bolus enter into a respiratory system. Is that right? It can even kill the person if a big bolus does enter there. So this should be prevented. Now how it is prevented? How do you prevent it? Yes. Number one, these vocal cords, swallowing reflex, swallowing reflex to stimulate and vocal cords will be strongly approximated. Vocal cord will come together. That is why you cannot swallow and talk simultaneously. Just imagine you are swallowing something and try to talk. Imagine you are swallowing something. Do you feel dumb or not? Try to talk. While you just imagine that you are swallowing something. Simple imagination activates the swallowing mechanism in such a way, vocal cord closed and you cannot talk. Is that right? So vocal cord will be approximated, but there are further safeguards also. What will happen? This whole larynx will move upward and forward. It will try to come under the tongue, try to get away from the root of the food bolus. You are understanding? Just hold your larynx and try to swallow your own saliva. Larynx moves or not? Where it goes? It goes upward and anteriorly. It is trying to come, it is trying to move in number one upward, number two interiorly. So it is trying to go under the tongue, trying to hide it, okay? Is that right? And when it will go there, what will happen to this lid? This will close on that. You are understanding? What is epiglottis will? Close on that. So what are the steps which have happened up to now? That as soon as food bolus at the end, once you feel that you have chewed enough and it is ready to be swallowed, with the tongue you press the food bolus against the hard palate and then it slips backward. And there it stimulates the swallowing reflex receptors, epithelial, especially on the tonsillar pillars and around that entry of the oropharynx. At that very point, right, fifth and ninth nerve take information to the central nervous system, swallowing center is activated and that stimulates multiple motor nerves which are going to bring changes in all this area. What are the changes? Soft palate lift upward, block the way to the narrow. What is this? Nasopharynx. Vocal cords approximate. Larynx move upward and forward. What is this funny thing? Yes, this will come over it, epiglottis. Is that right? Will come over it. And with that, another thing will happen. Listen, when larynx will move upward and interiorly, what will open here? Upper esophageal sphincter. Upper esophageal sphincter, remember one thing, before I move to the esophagus, I must tell you, upper esophageal sphincter is the lowermost part of inferior constrictor of the pharynx. You know, pharyngeal wall has three muscles. There's superior constrictor, is that right? Then there is middle constrictor and then there is inferior constrictor. And inferior constrictor has inferior most area which is connected with the cricoid bone. You know, under the larynx here should be which bone? Cricoid bone. So this lowermost part of inferior, what is this? Inferior pharyngeal muscle is cricopharyngeus muscle. Right? This is acting also as upper esophageal sphincter. When larynx move upward and interiorly, upper esophageal sphincter become open. Now you see you have closed the way to the nose, you have closed the way to the, what is this? To the airways. Another thing, actually when you are pushing the food backward, even the, you know, tonsillar pillars, palato-glossus muscles, right? They also approximate, so that they approximate, and when they muscles which are muscle arches down from here to the tongue, here on one side and other side, these muscles approximate and make a slit so that very big bolus should not go down. Sometimes you fail to swallow too big things. Is that true or not? Right? So, again, let me repeat and put it in proper perspective. As soon as swallowing reflex is stimulated, soft palate move upward, nasopharynx closed. Vocal cords approximated, larynx move upward and forward, and epiglottis covers over there, right? And now when larynx move forward and what happens? Upper esophageal sphincter open. Now food has only one way available. Almost at that very moment, what happens? That peristaltic wave is produced in pharyngeal muscles. Peristaltic wave, right? And that wave, you know, constrict above the food and relax below the bolus. So what will happen? Peristalsis will sweep over all pharyngeal muscles from superior pharyngeal muscle. First superior pharyngeal muscle will constrict, push the bolus downward, then middle pharyngeal muscle will contract, and then inferior pharyngeal muscle will contract and food bolus will enter into upper part of esophagus, and we say which part is completed? Pharyngeal phase of swallowing is completed. Am I clear? Now after the pharyngeal phase has been complete, remember it takes about 1 second, for this stage to push it from oral to pharyngeal side. And in the pharyngeal side, all this takes less than 6 seconds, right? Usually 2-3 seconds. Now, once the food is in upper esophagus, now esophageal stage of swallowing start. In upper esophageal stage of swallowing, we'll take the bolus from the upper part of esophagus up to the stomach. It takes about 8 to 10 seconds. And gravity helps us and it may become fast. Is that right? But remember that esophagus can swallow even in anti-gravity fashion. If you decide to eat in a very specific position, you first go upside down, hang it down and then try to eat, you can still eat it. Is that right? So it means esophagus can take anti-gravity also food, but it's better to take the help of gravity and eat in a proper position. Now, what happens when food bolus enters into esophagus? The law of gut will operate. What is that? That number one, this food bolus will stretch that part of the esophagus. Is that right? Then what will happen? Peristalsis should be produced. But remember, primary peristalsis is not produced by this way. Primary peristalsis in esophagus is, which initially takes the food bolus down. That is simply a continuation of pharyngeal peristalsis. Let me repeat it. As soon as bolus comes into upper esophagus, it does not produce a new peristalsis in the esophagus. No. Actually, the peristaltic movement which is sweeping over the pharynx, which has taken the bolus up to esophagus, same peristaltic constriction ring continues all over the esophagus. But you can understand that my enteric plexus in the esophagus is helping with the whole system. Is that right? That constriction ring is just, yes, what is happening? That as bolus is moving forward, right? Constriction ring is just proximal to the bolus, right? And relaxation, receptive relaxation is distal to the bolus. Am I clear? Now, as the food bolus is going down due to primary peristalsis, which was original continuation of the peristalsis from pharyngeal action, right? It will take the food bolus up to the stomach. But normally, lower part of the esophagus here is slightly thickened. And here smooth muscle. Here the smooth muscle has a higher tone. It has normally high tone and it remains closed. And so, if food bolus has to pass through this area, which is called lower esophageal sphincter, right? If food bolus has to pass through this area, then this must be relaxed. But you know the mechanism. As food bolus is approaching, the descending connections of my enteric plexus will produce inhibitory, what? Neurotransmitters here. VIP and nitric oxide. So actually, just above the bolus, acetylcholine and substance P is being produced, and below the bolus what is being produced? VIP and nitric oxide, and this will keep on pushing the bolus downward. So naturally when bolus is coming down, lower esophageal sphincter will relax. This is very important point, that approaching peristalsis relax the lower esophageal sphincter, not only lower esophageal sphincter but also the upper part of stomach. This is called receptive relaxation. That these part of gastrointestinal system are getting relaxed to receive the food. What we call it? Receptive relaxation. So when food bolus is coming near, lower esophageal sphincter automatically relax as stimulation to the enteric plexus generate lot of inhibitory neurotransmitter VIP and nitric oxide here, and when it relaxes, food bolus will come to the stomach. Am I clear? Now why I am stressing so much here? Now it is not happy because there is acid pool there. I am stressing so much that in some diseases, in some conditions, lower esophageal sphincter fail to relax. And if fail to relax, food will keep on accumulating in esophagus and it will not enter into stomach properly in time. Do you know a disease like this? This condition is called achalasia. Have you heard of achalasia? Right? What is achalasia? Achalasia is basically a problem in which, clinical condition in which lower esophageal sphincter fail to relax on anticipatory arrival of peristalsis. Normally it should. Is that right? We'll discuss about achalasia later. But of course you can understand now why achalasia should be there. Why the achalasia should be there? Very simple. Very simple. Tell me. Because enteric plexus is not working here. Because the duty of the enteric plexus is that as bolus is moving forward, it should produce a receptive relaxation in the distal segments of the gastrointestinal system. So in some diseases, some conditions, enteric ganglions are not present here. When enteric ganglia are not present here, congenitally, then this achalasia will be present, or difficulty in swallowing will be present from very beginning. Difficulty in swallowing is called dysphagia, right? Now, in achalasia, dysphagia occur, difficulty in swallowing occur. It may be in early part of the life or it may be secondarily. There is a condition, there is a parasite called Trypanosoma cruzi. Trypanosoma cruzi cruises down here, and I don't know why it loves to destroy the ganglion of enteric plexus, especially in this area. Trypanosoma cruzi. And when it destroys the ganglion here, you can understand what will happen. What will happen? Food will come down up to this, but because it failed to show any relaxation, right, because VIP is not there, nitric oxide is not there, so what will happen? Food will accumulate in esophagus. But here I want to tell you one thing more. In normal esophageal stage of swallowing, usually primary peristalsis start from pharyngeal side and continue down there. But sometimes you have eaten some kind of food that some of it may be sticking here and there. Let's suppose all the bolus has gone down, but one piece of the bolus is sticking here and refuse to go down. What will happen? Then other peristalsis will be produced only in this area and sweep down. And it will keep on sweeping until this monkey goes down, the sticking food. You are understanding? Those peristalsis are called secondary peristalsis. So normally esophagus has which type of peristalsis? Primary peristalsis, which is a continuation of pharyngeal peristalsis. But if some food is retained within the lumen of esophagus, wherever it is retained, secondary peristalsis are generated there by distension and stretch. Right? And they will keep on working downward until the retained food is thrown into stomach. But if achalasia of cardia is there, then what will happen? If achalasia is there, in achalasia, esophagus will become like this. It will become dilated at the top because you are bringing the food there. But here lower esophageal sphincter refuse to open. Is that right? And in this case, what will happen? If food is too much accumulated here, it will get infected, because normally when food goes down into stomach, if there are bacteria there, and of course food has a lot of bacteria, so they will be killed by the HCl. But if food is retained for a longer time into esophagus, does not have any effective mechanism to kill the bacteria, food gets infected and putrefied. So person will have very bad smell. But this is really not most dangerous thing. The most dangerous thing is when this person will sleep. Maybe this food enter into respiratory system. Because when this person will sleep, these reflexes will become slightly inhibited. And there's a chance a little piece of food goes down and disturb, or infection start in respiratory system. Right? And this person cannot eat well. Of course, he will be wasted. So achalasia is a condition in which lower esophageal sphincter fail to relax on arrival of the peristalsis in esophagus, and food, a larger amount of food collect into esophagus, even though this is not a mucosal disease. But when food remain here and become putrid and infected, it damages the mucosa also. And mucosa of esophagus also become what? Ulcerated. Right? That produces substernal pain also, and person says I cannot swallow well. Right? So anyway, so these were few words about your esophageal reflex. Do you have any question? There's no question up to this? Right, now we'll see that we have done this voluntary stage, which initiates the swallowing, is 1 second. Pharyngeal stage will take 3, 4, 5 seconds, but interrupts the respiratory system only for a very brief time. And esophageal stage takes how much time? Esophageal stage 8 to 10 seconds. Is that right? But gravity assist, right? And then never forget, when esophageal peristalsis is coming down, receptive relaxation is not only in lower esophageal sphincter, but it is also in upper part of stomach. Stomach gets ready to receive the. Okay, now we come to the. Okay, he wants me to have a break. Now we will talk about the motor functions of stomach. But remember, stomach has other functions like secretory functions and digestive functions, right? But right now we'll talk about motor functions of stomach. Right. You already must be knowing about the musculature of the stomach, right? In histology lectures you must have done that stomach has basically mucosa, submucosa and muscularis externa. And in muscularis externa, unlike other part of gastrointestinal system, it has 3 layers, 1 additional layer. Because other part of the gastrointestinal system has muscularis externa with outermost longitudinal layer of muscles, then inner, which is circular layer of muscle, but in the stomach there is an additional oblique layer of muscles inside the muscularis interna, innermost layer, right? Secondly, all the musculature in the stomach is smooth muscle. Why I am saying? Because if you compare it with the esophagus, upper 1/3 of the esophagus is striated muscles. Middle 1/3 of the esophagus is striated and smooth muscle, and lower 1/3 of the esophagus is smooth muscle. But stomach all striated muscles. Now, when food, what are the 3 basic functions of the stomach? 3 basic functions of the stomach are: number 1, storage, motor function. Number 2 is mixing movement, and next is the propulsive movement, but actually propulsive movement help the stomach to empty itself, right? So we call it, yes, what? Emptying of stomach. Of stomach. Okay, let me make a smaller muscle, then we talk about its different features. Anatomy of the stomach, basic things you must be knowing, that stomach is divided into which 3 anatomical regions? Yeah, this uppermost is fundus, right? Then here is the body, and here it is pylorus, antrum, body, and then there is antrum, and last part of the antrum, which is the exit from the stomach, that is called pylorus. Is that right? So there is fundus of the stomach, body of the stomach, antrum of the stomach, and the exit point from stomach to the duodenum is called pylorus. Is that right? And this area of the stomach, where it is connected with esophagus, there is lower esophageal sphincter, which is normally closed to prevent the reflux. And normally, the smooth muscle layer in pylorus is also thickened and tonically almost closed. Is that right? Now, when we are eating, food is initially stored in stomach, right, so that it can be gradually released as food is being processed by the small intestine, by secretion and absorption, right? Now, for this storage purpose, when food comes from esophagus here, I told you in the last section that when food is reaching here, due to my enteric plexus, right, stimulation, this part of the stomach will undergo receptive relaxation. What kind of relaxation will be there? Receptive relaxation. But as more and more food come, stomach keep on relaxing, right? The second type of relaxation is called accommodative relaxation. Receptive relaxation is that, let me differentiate. Receptive relaxation is when Peristalsis from esophagus is coming down, and along with the relaxation of lower esophageal sphincter, upper part of stomach also relaxes. Right? That is called receptive relaxation, and that is through the enteric plexus. Right? But actually, when food comes over here, and more food is reaching stomach, do you think pressure in the stomach goes up or down or remain the same? It remains almost same, because within physiological limit, if you're eating within physiological limit, right, then what happens as you keep on eating, more and more volume is coming here, right? That volume stretches the, what is this? Stomach wall, and that stretch activate sensory fibers through the vagus nerve to the central nervous system, and then from there fibers come and relax, motor fibers come back and relax the stomach smooth muscle. So actually stomach keep on increasing its capacity, of course within limits. So what happens as you are dumping more and more food in your own stomach, right? Not only there is receptive relaxation, there is also accommodative relaxation, that stomach is going to accommodate more food. This accommodative relaxation is not a local reflex, it is vagal reflex, because after sensory pathways through the vagus as well as motor pathways through the vagus. Are you understanding me? Now what happens when more volume is there, a little stretch on stomach wall will actually produce action potential in the sensory fibers in the vagus. They will take information to the where? Medulla. Is that right? They will take the information medulla, and from there the parasympathetic fibers, motor fibers which come to the stomach, their action will be reduced. And you know, parasympathetic is motor to the stomach and GIT. When their action potentials are reduced, the less stimulation of the whole smooth muscle and enteric plexus of stomach, and stomach get relaxed. Is that right? This is called accommodative relaxation. Is that right? Normally, stomach can store from 1 liter to 1.5 liter. Of course, exceptions are there, but usually human stomach can take 1 liter to 1.5 liter of food over there. Is that right? Now we come to the, once the food is there, right, then how the mixing movement start. Right now I'm going to talk about the mixing movement. I'm talking about this thing. Actually, in previous lectures I've told that in this part of the stomach there are special cells which are called interstitial cells of Kegel. Interstitial cells of Kegel. They act like a pacemaker, as there is SA node in the heart, right? And in 1 minute they produce around 3 to 4 slow waves. Is that right? And usually those slow waves cannot produce significant contractility, but at the top of the slow waves, if there are spike potentials, we have discussed in previous videos, and that will produce effective contraction here. Now there are two types of contractions which are produced in upper mid section of the stomach, right? Every 15 to 20 seconds. Is that right? Contractions which are produced, they are about 3 contractions per minute, because the slow wave basic rhythm of the stomach is, slow wave electrical activity in the stomach is at the max 3 per minute. Is that right? Now, when the contractions start here, what you need to remember, there can be weak contraction and there can be strong contraction. Now weak contractions are mixing contractions, and strong contractions will help in emptying. Now let's talk about the weak contraction. How the weak contractions work. Actually, when weak contractions are produced, they will compress stomach here and here. Is that right? From both sides. Now it will produce a slight indentation. This part of the wall of stomach will dig into content of the food. This will dig in, then this constriction ring, a sort of, you can say constriction ring is produced here, here and here. Now this narrow area constriction ring will move downward and distally. You know the law of the gut. Is that right? Same principles of enteric plexus, right? Now, every minute, around 3 constriction rings are produced, and these constriction rings, initially which are weak constriction ring, they move downward and towards the pylorus, right, from the body to the antrum to the pylorus, and they take the content of stomach forward. Now what will happen to pylorus? Yes, ma'am. What will happen to pylorus? It will open. What do you think? Inhibited, yes. Yes, Priya. If this is what type of contraction? Weak contractions are there. Every minute about 3 are produced, and they indent like a constriction ring into the content of the food, and they're moving, sweeping towards from middle of the body to the antrum to the pylorus. What happens to pyloric ring? It does not open. It's not everywhere that some constriction come and it should open. There are exceptions to the laws also. This is one exception. When mixing is going on, it does not open, rather it becomes a little more tight. Now you may be thinking, what is this thing? But this is a very important thing. You know what happened? Look here. Let's suppose, according to your theory, look here, which is wrong, that if it contract and it move down and it open, all 1.5 liter will go to duodenum. Can duodenum handle that much? No. So what really happens when stomach weak mixing movements start and they move downward, it becomes even more tight. So all the content of stomach which are sweeping towards here, they will hit it and fall back. What will happen? That these contents of the stomach which are moving right ahead of this, they are moving like this, only those particles which are very small, because it may be very narrow, it becomes more narrow, right? They will all hit back. And this activity keep on doing for a long time. So what will happen? All the contents of the food will get mixed with the gastric secretion. This is called enteral pump, because originally these waves become very prominent in the enteral region. They start very weak or feeble from the body. As they move downward, they become slightly more prominent, and their velocity becomes fast and intensity becomes more. So what is happening? That these constriction rings, which are relatively weak, as they move towards the antrum and pylorus, they progressively will become slightly strong and slightly fast, and contents are splashed against the semi-closed pylorus, and very little material goes to duodenum, right? Most of it will be, what happened? It will fall back. It will not go into duodenum. Is that right? Now, this is a mixing mechanism. Stomach is acting as a mixer. It is mixing, right? And this movement of food is called retropulsion. Retropulsion. Look, when this ring starts moving, there is propulsion of food. When food reaches here and splashes back, that is retropulsion. So this is what happens in stomach when it is mixing its contents with the gastric juices. Any question up to this? And how many movements are there? About 3 movements or 4 movements per minute. Is that right? Now, after we have discussed the mixing movement, advantage of mixing movement is that for long time it will keep on doing like this until the contents of the stomach, the food particles, water and secretions, they become semi-solid paste-like material. When they become something like a paste, then only that paste will be emptied here. That paste is called, what is it called? Chyme, C-H-Y-M-E, chyme. So stomach actually convert whatever you have eaten deliciously with very different colors and different type of combinations in the stomach. Eventually, due to its mixing movement, it will become chyme. And chyme is a paste-like material, a semi-solid, semi-fluid material, depending upon the proportion of water and the solid component, and secretions from the with the gastric glands, right? This chyme will be gradually moving forward. Now, this mixer is very important, because every time when these mixing movements are approaching, rather than opening, it becomes a little more tight, so only very fine particles will move forward. Do you think there's a chance to too much overload that duodenum? No. Is that right? So this was something about which movement? Mixing movement. And now we come to after the mixing movement, Okay, before I go to the emptying movement, I want to explain another thing. Okay, let me explain first emptying movement. There's not much difference. Emptying movements are those movements in which these constriction ring becomes stronger. And this time pylorus does not close significantly. It still closes but not very narrow. So actually in the stomach, 80% of the time there are mixing movements or weak peristalsis. And 20% of the time there are strong peristalsis. So few mixing movement, then one very strong peristalsis come and move this content very strongly and splashes against the pyloric sphincter, and that moment pyloric sphincter is a little open. So if some paste is present, some paste has been made, that will be pushed into duodenum. Am I clear? So what is the main difference in mixing movement and the propulsive movement of or emptying of the stomach? That mixing movements are weak and they are 80% of the time. And propulsive movement or movement which empty the stomach, right, which are called enteral pump, right? These movements are strong and they are 20% of the time. Is that right? And all this mechanism ensures that only chyme should move to the duodenum. Any question up to this? There's no question. Now, another type of movement I want to mention, that when you are fasting, a hormone is produced called motilin. What is that called? Motilin. This can produce special type of movements almost every 90 minutes. When your stomach is empty or you're fasting, right, motilin hormone goes up in your blood and this acts on the smooth muscles of stomach and it moves the stomach. Sometimes it produces rapid movement in the body of stomach, rapid peristalsis in body of stomach, and those are called MMC. What is MMC? Migratory myoelectric complex. Migratory myoelectric complex. Don't tell anyone this is mixing movement or it is emptying movement. No, it is usually an empty stomach. Is that right? Migratory myoelectric complex, right? This is produced by motilin. This is a hormone, right? Produced by the small gut. Another thing which I want to tell you about the hunger contractions. When you are hungry for a long time, right, what really happens that their rhythmic contractions start in stomach. Right? First of all, before I go for that, what is the function of this thing? What is the function of this thing? They are looking for any residual food to push forward. That if still there is something, right, push it forward, right? Now hunger movements. Hunger movement in stomach occurs when you have been fasting for significant time, and this movement start peristaltic movement rapid in the body of the stomach, right? And sometimes there are multiple movements started and it go into tetanic contraction. You're getting, stomach is getting angry because there is no food. Is that right? And it may produce even a little pain in the pit of this area. What is that called? Hunger pangs. P-A-N-G-S. Hunger pangs. Is that right? So, when stomach is full, it moves. When it is very empty, it also moves. And hunger pangs start about 12 to 24 hours after the last meal. And within 3 to 4 days, it reaches to peak and then gradually dies out. And remember, hunger pangs or the hunger contractions, and when they become painful, it is hunger pangs. They are more strongly felt in young and healthy people because their GIG tone is high. Or hunger pangs are more strong when you have hypoglycemia. Am I clear? There's no problem up to this. Now,
what causes hunger pangs? Like what causes the actual...
that is electrical activity. That is not related with the motilin.
Okay.
Motilin, right? Now we come to how the pyloric sphincter open. Right now I'm going to talk about that thing. Right now I'm going to talk what are the factors which determine the stomach emptying. You know who should control the rate at which stomach empty itself? Who should control? Who is receiving this stuff? Duodenum. Duodenum is narrow. So duodenum should have some mechanism to control the stomach so that stomach does not become stupid and put everything there. You are understanding? So the real powerful control of emptying mechanism of the, what is this? Stomach is mainly by the neural and hormonal mechanism from duodenum and jejunum. Because duodenum and jejunum are dealing with the chyme which is coming, and they have to properly digest the chyme and absorb the chyme. And if too much stuff come here inappropriately, too fast, is that right? Can proper digestion and absorption occur? No. So actually the most powerful regulation of stomach emptying mechanism is by the duodenum. It means duodenum and jejunum should have some mechanism to control the emptying. And how to control the emptying? Number one, it should be able to control the movement of these strong peristalsis, plus it should be able to control the pyloric sphincter. Is that right? Now, let me explain under what circumstances, what are the factors which regulate the emptying of the stomach. As I told you, more powerful factors are from duodenum, but the weak factors are from the stomach itself. Right. Now, here is your pyloric sphincter. This is lower esophagus. Okay, now, I'm going to talk about the factors which determine how fast or slow stomach will empty its contents into duodenum. There are gastric factors which are weak, and there are duodenal factors which are powerful. Let's talk first gastric factors. Number one, of course, volume of the food. When you increase the volume of food, the rate of emptying slightly increases. Is that right? How? Why? What is the mechanism? That volume of the food is more, then rate of the emptying will slightly increase. What is the mechanism? Yes. Do you think when there is more food, it will put more pressure and that pressure will drive? No. Remember, when you are putting within physiological limit, when you are putting more food, actually stomach relaxes and pressure in the stomach does not go up. Actually what happens, even though stomach relaxes, but stretch on the wall become more. So when you are eating more, you are putting more volume into your stomach, pressure in the stomach does not go significantly high, but stretch on the wall of the stomach goes significantly high. The more stomach stretches, smooth muscles more open, their calcium channels open, and they produce stronger contractions. And when there are stronger contractions, rate of emptying becomes slightly more. Is that right? So one gastric factor is volume of food. Right? Second factor is that stomach produces gastrin. You know, gastrin is a hormone which is produced by some specialized cell in antrum. Right? G cells in antrum, they can produce gastrin. Primarily, gastrin is produced to stimulate the HCl secretion from the gastric glands. But some people believe that gastrin also slightly increases the intensity and velocity of stomach contraction. And of course, you must be knowing that parasympathetic nervous system stimulates the stomach or inhibit the stomach. Parasympathetic nervous system. Acetylcholine, of course, stimulates the stomach. Is that right? Because if someone is having a drug which is anticholinergic, stomach will not contract well. Any question after this? But really, the thing which you should understand very clearly is duodenal factor. Right? Duodenal factor and stomach emptying. Let's talk step by step how the duodenum control the stomach motor activity. Number one, let us suppose that from the stomach too much contents come here. It is overfilled. What is overfilled? Duodenum. It should slow down or accelerate stomach. Slow down naturally. Now, there are three levels of mechanism by the nervous system. Number one, there are, whenever certain stimuli come to the duodenum, stomach motor activity will be inhibited. Now these are neuronal. So we call this gastric reflex. What reflex? Enterogastric reflex. It is not reflux, it is reflex, neuronal. This inhibits the stomach. Exactly how? There are 3 levels of enterogastric reflex. Number 1, that when too much food come here, chyme come here, and it is stretched, this area is stretched, that will produce inhibitory neurotransmitters on the back. Through which mechanism? What is this mechanism? Enteric reflex. Because enteric reflex is present throughout the gastrointestinal tract. So when duodenum become overstretched or there are certain stimuli which come to the duodenum, what duodenum will do? Neuronal response. What is neuronal response? That stretch receptors in the duodenum will activate the ascending pathways of enteric system, which will produce inhibitory neurotransmitters in stomach. And stomach movement will become slow and it will not dump further chyme into duodenum until duodenum deal with those situations. Am I clear? Secondly, another way. Not only this is local reflex, another reflex which is seen there is that some sensory fibers from the duodenum, they go up to sympathetic ganglion. Suppose here is central nervous system and this is sympathetic preganglionic fiber and here is sympathetic postganglionic fiber, and let us suppose these are some sensory fibers. Now some sensory fiber from duodenum, they go up to the ganglion. Which ganglion? Sympathetic ganglion. Right? And in the sympathetic ganglion, they stimulate fibers which are going back not to duodenum but going back to stomach. And when sympathetic activity of stomach is increased, motor activity of stomach will be inhibited. Is that right? So it means up to now I've told you 2 reflexes. That duodenum through the enteric reflex can inhibit motility of stomach, and duodenum through the ganglionic reflex, through the sympathetic prevertebral sympathetic ganglion, it can lead to inhibition of stomach. And still, if it's really very much irritated, more sympathetic fibers are stimulated and these fibers will take the information through the vagus. Sensory fibers in the vagus nerve, they will take to the central nervous system, and from there vagus fiber will come and inhibit the stomach. Now the reflex has become very long. The reflex has gone through central control. So duodenum knows how to slow down the stomach through local enteric reflexes, through reflexes through the sympathetic prevertebral ganglion, or vagovagal reflexes, which are initiated from the duodenum and inhibit the stomach. Am I clear? Now question is that what are the circumstances which activate this reflex? Which reflex? Enterogastric reflex. One thing is very simple, that simply whenever there is over distension of duodenum, these reflexes will be activated, right? But duodenum has many chemosensors, right? Osmoreceptors, chemoreceptors. So even if it is irritated by any means, duodenum has irritation, right? It will inhibit the stomach motility, movements of motor activity of stomach. If there is too much protons coming here, for example, so much chyme is coming which is highly acidic, right? For example, pH in the duodenum goes down up to 3 or 3.5. Duodenum is not happy with it, so to want to neutralize the acid. So to buy the time, high acidity will stimulate the receptors in the duodenum mucosa and inhibit the stomach until bicarbonate in pancreatic juice and bile and neutralize this acid. Even if chyme which is coming, it is not isotonic, it is hyperosmotic, osmotic, or very much hypoosmotic. If such type of chyme come, is that right? where osmolality of the chyme is very much different from our body fluids osmolality, it will immediately inhibit that. You know why? Imagine that if very much hypertonic drink comes on this pathway and GIT gets fully loaded with hypertonic drink, what it will do? It will absorb the water from mucosa. Is that right? And reabsorption will not occur because hypertonic fluid sucks the water from everywhere. Are you understanding me? And that will produce hypovolemia, fluid electrolyte changes in our whole system. Is that right? Or if very hypotonic fluid comes, this fluid will very rapidly absorb, and again fluid electrolyte shifts will occur. So duodenum has osmoreceptors also, right? So what it does, that if very hyperosmotic or hypoosmotic fluids come, it will slow down the stomach. So the stomach keep on producing juices and try to bring the osmolality near to body fluids. And it deals that kind of fluid very gradually. Are you understanding what I'm saying? Right? Then, even if there are breakdown product of protein or fat digestion. If too much protein digestion, breakdown product come too much, right? Improperly digested, not properly digested, it will slow down because it knows it needs to digest them and it will take time. Or if too much fat come here, right, that can also irritate the mucosa, it will slow down. Why? Because digestion of fat, you need pancreatic lipases and bicarbonate and bile salts to emulsify. So duodenum and jejunum have to work a lot on the fat, so they slow down the stomach. Am I clear? That is why when you take food rich in fat, stomach emptying become very slow and digestion process become very slow. But if you take it in protein, rich in protein, it will be little faster. And if you take it carbohydrate, it will be more faster. That is why when you take too much fat in the food, you say it's a heavy food. You are understanding? Because duodenum want to work and jejunum longer time to digest the fat and absorb the fat. So they just slow down the stomach and it become heavy over there. Am I clear? So these are the stimuli: distension, irritation, high acidity, too much hypertonic or hypotonic fluid, chyme, or too much partially digested proteins or fat. Right? They will activate the enterogastric reflex, which may be local reflex or through the sympathetic ganglion or through the vagovagal reflex. All of these will slow down the stomach movement, right? Plus tighten the pylorus. So pyloric sphincter become tight and stomach emptying will become slow, right? Opposite to that, of course, if there is too little chyme, of course reverse will occur. If isotonic fluid is coming, slowing down will not occur. You are understanding that? Yes.
Which of those three pathways does CCK run?
I'm going to talk about that. So this was about neuronal control through the neuronal system. Duodenum and jejunum control the stomach. Any question up to this? Then there is hormonal control also, right? There is hormonal control also. Let me explain. Not only neuronal control, but hormonal control also. For example, if too much acidity come, okay, first of all, if too much fat come, right? Number one, through enterogastric reflex, it will slow down. Secondly, there are special cells here, eye cells, and they will produce what? Cholecystokinin. They will produce a hormone called cholecystokinin. Now, cholecystokinin has primarily two functions. Number one, cholecystokinin will go to the gallbladder and gallbladder will contract. Cholecystokinin. Cholecyst is what? Gallbladder. Kinin means movement. So gallbladder moving hormone. So basically original function which was discovered of this hormone, it contracts the gallbladder and bring lot of bile here with bicarbonates. Plus cholecystokinin also stimulate the acini and the exocrine pancreas and bring lot of pancreatic enzymes including lipases. So it can, bile acids come here, pancreatic lipases come here, so they can digest the fat. But because this process takes long time, especially pancreatic lipases work in alkaline environment, so we need to slow down the stomach. So cholecystokinin also has a receptor on stomach, smooth muscle, and inhibit them. Is that right? So that is why when you eat very fatty food. It takes long time to digest. One simple reason is that fats will float in stomach, so they will take delayed time to reach here. And once they reach here, they slow down the stomach further. Am I clear? Then, I'm talking about now hormonal control. Duodenum and jejunum control the stomach hormonally as well as neuronally. Now hormonally, if too much protons come here, then there are S cells here which will produce secretin. Primary function of the secretin is that it will activate the pancreatic ductal cell to produce more bicarbonate. So that what should happen? Neutralization of the acid. But secretin also act on stomach and slows down the stomach. So cholecystokinin, this also slows down the stomach. Secretin also slows down the stomach. Because in both cases, duodenum and jejunum are buying the time. Is that right? In first case, to digest the fat. In second case, to neutralize the acidic content. And then, if fat and carbohydrates both come together too much, fat plus carb, another hormone is produced, and that is called GIP, gastric inhibiting peptide. Of course, this name is telling that it will inhibit the stomach. Gastric inhibiting peptide. Am I clear? Any question up to this? Now, how to remember all these factors? It's very easy. One thing you know, parasympathetic system stimulates the stomach and sympathetic system, yes? Sympathetic system inhibits. If you know this thing, let's remember it like this, that parasympathetic nervous system moves gastric, yes, emptying, or gastric system. Now, parasympathetic move for motilin. Gastric means for gastrin. So these are three stimulating mechanisms. If you remember, parasympathetic nervous system moves the stomach, moves the gastric system. So actually three things. Parasympathetic nervous system, cholinergic, plus move for motilin, plus gastric system for gastrin. Right? But now you remember other thing. Even though parasympathetic nervous system moves gastric system, but sympathetic nervous system secretly, yes, inhibits gastric system. Do you think it's difficult to remember? Now what is here? Sympathetic nervous system also inhibits. Secretly, secretin also inhibits. Is that right? And gastric inhibitor, GIP, also inhibits. Now what is left one to remember is cholecystokinin. So sympathetic nervous system secretly inhibit gastric system for cholecystokinin. That's right, for cholecystokinin. So most of the questions which they form on stomach are movement of the stomach, that they are mostly concerned that do you know the mechanism of emptying of stomach and how the duodenum regulate that? Is that right? So if I asked you enterogastric reflex at 3 levels, number 1 local enteric, number 2 through sympathetic ganglia, number 3 vagovagal through central system. And if we talk about hormonal control, what are those control? If there's too much fat coming, cholecystokinin is going to inhibit. If too much protons come, secretin is going to inhibit. And if fat and carb both come, GIP is going to inhibit. Is that right? Now, very briefly about the pyloric stenosis. Sometimes there's pathological narrowing of pyloric area. We call it pyloric stenosis. Pyloric stenosis, narrowing of. This may be due to congenital. In some, especially, it's more common in male infant. They are born with hypertrophied or extra thick layer of pyloric muscle. right? So newborn males or male infants, they may develop pyloric stenosis. Or pyloric stenosis may be seen in those patients who have peptic ulcer for a long time. And peptic ulcer lead to inflammation and scarring. If the scarring is very near to this area, this scar become contracted and this area become narrow. So inflammation and scarring of pyloric region due to peptic ulcer disease. That will also produce stenosis. Thirdly, unfortunately, someone develop cancer in this area, right? Malignancy. And that makes this area narrowing. So these are all different causes of pyloric stenosis. And usually what happens in pyloric stenosis, when this area becomes narrow, can stomach push the food forward or climb forward? So basically, person, whatever he eats, person will vomit out. And specific thing in this will be, this vomiting will not have bile. Because bile is coming to duodenum. And content of duodenum in this type of vomiting cannot come back because there is pyloric stenosis. So we call it non-bilious vomiting. The vomiting which is produced in these patients is non-bilious. Bile is not there. Any question up to this? Then another condition which is called gastroparesis. Gastroparesis is that stomach becomes semi-paralyzed and it becomes very relaxed and it takes very long hours to empty itself. Gastroparesis. What will happen to a patient with gastroparesis? What will happen to you if your stomach keep on receiving the food and does not pass it forward? You will develop abdominal discomfort in this area. You will develop nausea and vomiting. If you cannot move it forward, you may throw it out, throw it up. Is that right? So nausea and vomiting. You may develop sensation of balloting here. You may develop sensation of satiety. You eat very little and feel you have eaten enough because your stomach is all the time full. Is that right? These situations are seen in different mechanisms of gastroparesis. Any question? Class dismissed. Right. Today we are going to talk about the movement of small gut. Right? Movement of small intestine. Small intestine. Small, you know that small intestine consists of 3 parts. Yes, what are those 3 parts? Yes. Yes, you start with the duodenum. And then there is, yes, jejunum. And then we have ileum. Is that right? And ileum terminate at cecum. Now, here we have duodenum from here up to here. Here is jejunum. About 5% of small intestine is, what is this? Duodenum. About 45% is, or yes, 40% is jejunum, and about 55% is ileum. Right? Now we are going to talk about the movements. First of all, what is the purpose of small intestine? The major purpose of small intestine is digestion and absorption. Right? So it means the movement which are going to be there in the small intestine, they must serve and help in digestion and absorption. And the motility in small intestine, what it is doing there, the movement in small intestine. Number 1, it moves the content. Right? It moves the content throughout. And for movement purpose, of course, the movement generated a propulsive movement which are peristalsis. Secondly, not only we need to move the content through the small intestine, but we also need to mix the content of small intestine. We need to mix the contents of small intestine. Right? With what? With the gastrointestinal secretions. Because in small intestine, of course, from here, right, there are biliary secretions from gallbladder coming, and there are pancreatic secretions. Right? Small intestine is receiving the bile. It is also receiving the pancreatic juices. It is also receiving the intestinal secretions from the mucosa of the small gut, which is called succus entericus. So it is receiving bile, it is receiving pancreatic juices, and it is from its own wall. There are glands which are secreting small intestinal secretions, or which are called succus entericus. So all bile and pancreatic juices in succus entericus needs to be mixed with the content of small intestine, needs to be mixed thoroughly with the chyme so that digestion can be done well. Is that right? And another purpose of small intestinal movement is, of course, not only to move the content, not only to mix the content with the secretions, but there's one more purpose. Yes, what is that purpose? Just imagine. What could be the third purpose? One is very clear, movement of the, you are moving the content through peristaltic movement, right? Then you are mixing the content with the segmentational movement, or we'll talk about mixing movement. Do you think there's any other purpose of movements of small intestine? Yeah, they help in absorption, because when the contents of small intestine are churned, is that right? Then more and more areas of the content are exposed to the mucosal wall, so that mucosal wall can absorb the digested material, right? So another is that exposure of the content to the mucosal, yes, wall for better absorption, right? So what's there? That small intestinal movement move the contents, there should be peristaltic movement, and there should be mixing movement to mix the contents or chyme with the secretions, and of course moving the contents and churning the content in such a way that more and more contents of small intestine come in contact with the wall of mucosa, so that there should be absorption of digested material. Is that right? And of course sometimes small intestinal movement, they become reverse anti-peristalsis, which contribute to the vomiting. Is that right? So they also play a role in vomiting. Now, after knowing this thing, we know that small intestine has two types of muscles. Number one, there is muscularis mucosae in the mucosa, right? And there is muscularis externa, right? First of all, the major movement are not produced by the muscularis mucosae, right? The muscularis layer, which is in the mucosa, it produces only very minor changes in the mucosal lining, right? Major movements are produced by muscularis externa, circular layer and longitudinal layer in muscularis externa, which is a thick layer of muscles. But muscularis mucosae, which is a very thin sheet of muscles, that only produces different kinds of mucosal fold, right? They produce mucosal fold so that they increase the absorption and they increase the surface area for the absorption. Secondly, muscularis mucosae has also its smooth muscles going into the core of the villi, small intestinal villi, and those muscles elongate and shorten the intestinal villi, so that intestinal villi protrude deeply into lumen and regress back, and then deeply protrude into lumen and regress back, so that whatever in the lacteals, which are the lymphatic drainage of these villi, lacteals, in the lacteals whatever fats are there, they are milked and absorbed. So we also call it milking process. So muscularis mucosa has what kind of two activities? Number one, muscularis mucosa, which is controlled by submucosal plexus. Muscularis mucosa is controlled by submucosal plexus, point number one. Secondly, muscularis mucosa produce different kinds of infolding of mucosa in the lumen, so that surface area should be increased. Number three, muscularis mucosa is moving the villi, and because it is moving the villi, so it not only repeatedly villi get elongated and then shortened, elongated and then shortened, and in this way not only it helps in absorption, but it also milk the lacteals, so that whatever material is absorbed in capillaries and lacteals, that can be pushed into, or you can say flushed into drainage of vascular system. Am I clear? But muscularis mucosae don't play any major role in major mixing movement, and they don't play any major role in peristaltic movement, right? Another concept which is very important to understand, that usually it takes 3 hours to 5 hours for the chyme, right, or contents of the small gut to move from the duodenum and jejunum up to the ileocecal valve, right? It takes about 3 to 5 hours, right, to move the contents of small intestine from duodenum through jejunum, ileum up to ileocecal valve. Now the question is that sometimes it becomes fast and other time it becomes slow. What could be the reason? Can you tell me some factor? Sometimes whatever content are here, they will reach there in 2 hours, and sometimes it takes up to 4-5 hours. What could be the reason? Very good. It depends on the nutrients present in the chyme. If in the chyme there are more nutrients, it means we need more time for digestion and absorption. So if chyme is very rich in nutrients, this movement will become slow. But if chyme or the contents of the small intestine are poor in nutrients, we don't need much digestion and absorption, of course this will be faster movement, right? And of course you are very right that the nutrients which can maximum slow down the movement of small intestine are fat. You must be remembering that when fat is reaching to the duodenum, it activates the release of cholecystokinin, right? And cholecystokinin can modulate the small intestinal and gastric movement. Am I clear? Any question up to this? Right. Now we come to that basically small intestinal movement can be divided primarily into two. They are mixing movements, yes, and they are propulsive movement. Propulsive movement. Mixing movements are segmentation movement. Segmentation movement. And propulsive movements are peristalsis. We have already discussed in previous lectures about these two things. I will very briefly just review that segmentation movements are primarily produced by the circular muscles layer in the muscularis externa. And segmentation movement produces, as I explained previously, that if this is small intestine, it is at the moment number 1, right? At moment a little after that, it may divide into multiple segments due to constriction areas produced by the constriction of the circular layer, right? And after that, as I explained in previous video, that after a few seconds, 15 to 20 seconds, these constriction rings which are produced, they get relaxed and smooth muscle contract at other areas. And when it contracts at other area, then naturally what happen, that new constriction ring appear at new sites, right? And in this way, these contents which were present in one area, they will be chopped down into two. So what really happens, that such kind of movements which are called segmental movement or which are called mixing movement, they repeatedly appear in small intestine, and these movements are produced by which muscles? Circular muscles, and simultaneously in multiple areas of small gut, almost regularly spaced, constriction ring appear, right? And then those areas become relaxed and new set of constriction ring appear, and so forth. And this repeated action mixes the contents of the gastrointestinal system with the secretions, and also repeatedly expose these content with the mucosal wall. Is that right? And help in digestion and even absorption, right? By chopping these contents first here and then there, right? Like that. Now, the maximum rate of these movement really depends on what is the basic electrical rhythm of small intestine. As we mentioned previously, that in the duodenum, basic electrical rate or slow waves are produced at the rate of about 12 per minute. And as you move forward in the small gut, movements become slightly slow. So here it may be 9 or 8 slow waves per minute. So the maximum mixing movements, right? The maximum mixing movement per minute depends on what is the underlying inherent slow wave production rate. What is the basic electric rhythm. But usually, these segmentation occur 2 to 3 times per minute. They don't occur so much like 9 or 10 or 12 times. Full maximum frequency of segmentational movement is achieved when there is a strong stimulus, like when there is over distension of small intestine. A lot of chyme is coming over there. Do you have any question? Right. Now, this was about so we can say that small intestine act by virtue of small segmental movement act as a chopper every 2 to 3 times per minute, right? And now we come to what are another factor. If we give atropine, these movements become very weak. What does it mean? Or almost disappear. It means that without the functional activity of the enteric plexus, these movements cannot work. So what is the regulation of these movements, segmental movement? Enteric plexus, cholinergic fibers in the enteric plexus. Am I clear? Now we come to the peristalsis. Of course, you know what are peristalsis. Peristalsis are the movement which are generated in small intestine. Initially, at one point, a ring is produced, constriction ring is produced. Usually, the constriction ring is produced at the point where there is stimulus for the peristalsis. Stimulus means either there is stimulus from mucosal size, there mucosal area, like there are some chemoreceptors which detect presence of some contents within the lumen of the GIT, or there is some irritation to the mucosa. They can initiate the peristaltic movement, or when there is too much distension and stretch, receptors can also activate the peristaltic movement. And peristaltic movements are primarily controlled by the enteric plexus. Because they are controlled by enteric plexus, again, as we mentioned previously, when one part of the gut is stretched out or distended, is that right? Stimulus is there. Then on the ascending part, there are stimulatory action by release of acetylcholine and substance P. And on the descending side, there is inhibition by release of VIP and nitric oxide. So what really happens, that constriction ring, due to stimulation of oral side of the GIT appears, and this constriction ring, right, is simultaneously associated with development of relaxation on distal part. So contents of the GIT move aborally, but at new point again they give a stimulus and constriction ring move aborally, and contents again also move aborally, right? So this is how peristalsis in the small intestine move the content. Now these peristalsis again are produced with the maximum they can be produced at the basic electrical rhythm. What was that? Again, it was about 9 to 12 per minute in the duodenum, and it was 8 to 9 per minute in the ileum. What really happens, that on the slow waves, spike potentials start appearing, and that start generating the peristalsis. But again, never forget that those for peristalsis we also need enteric nervous system, especially my enteric nervous system. Again, if you atropinize the patient, you give the atropine, peristalsis will die out. Am I clear? Any question here? Right. Now, not only these peristalsis or small intestinal movement are controlled by the nervous system, local nervous system, they are also influenced by the extrinsic autonomic fibers. If there is parasympathetic stimulation, of course these movements become more intense and more frequent. And if there is stimulation of sympathetic nervous system, the small intestinal movement will become less intense and less frequent. So there is intrinsic neuronal control which is through the enteric system, and there is extrinsic neuronal control which is through the sympathetic and parasympathetic or autonomic control. Then hormonal control is also there for small intestinal movement also. For example, small intestinal movement are increased by gastrin, increased by cholecystokinin, and of course, if you want to remember only one thing, then you remember small intestinal movement are increased by? No. If you have to remember one single thing which can stimulate significantly small intestinal movement, motilin. Don't forget it. Motilin for movement. So we are talking about what stimulates the small intestinal movement. Of course, you have acetylcholine, everyone knows. There's no fun in telling. The parasympathetic nervous system motor to small intestine, and rather all the GIT and parasympathetic nervous system fibers do release acetylcholine there. But hormonally, if we talk about gastrin, cholecystokinin, motilin, and 5-hydroxytryptamine, which is also called serotonin. All of these are stimulant for the movement of small intestine. But inhibitors of movement of small intestine is secretin and glucagon. Right? Secretin and glucagon. When secretin is produced here, when lot of acidic chyme is coming, that is the moment to not to rush this material very rapidly forward. We want to slow it down and bring enough bicarbonate to neutralize the acid. You get it? So secretin also slows down the small intestinal movement. Secondly, glucagon. That is again logical. When the glucagon is produced, when you have very low glucose level, right? And when glucose level is low, it means GIT is empty. And what is the fun to mixing the secretions of the GIT with the secretions of GIT? There's nothing to get out of that. So movement should become intense or slow. So usually with glucagon release, movements in small intestine become slow. Then another thing about the movement of small intestine is gastroileal reflex. When you take a decent meal and stomach becomes stretched, right? Then when stomach becomes stretched or distended, it increases the movement in small intestine. Why? Because new meal has arrived, so let's move the things which are already there, move more distally. Is that right? So this is called which reflex? Gastroileal reflex, right? Or gastroenteric reflex. Now this gastroileal reflex, this is increasing the movement of ileum, right? So that ileum can push the material into cecum, and then jejunal material should come into ileum, and of course then duodenal material should come to jejunum, and if duodenum becomes empty, it gets ready to receive more chyme from the stomach. Am I clear? So gastroileal reflex, of course this reflex not only works through the enteric nervous system, my enteric plexus, but more importantly it works through the ganglions. I told you last time that from the stomach sensation will go through what fibers? They will go to autonomic sensory fibers to the central nervous system, and then from there parasympathetic fibers will come. It can be vagovagal reflex, or it can be just fibers from here, sensory fibers, number one, going to the brainstem, and then from medulla coming back here, vagovagal reflex. Or second thing is sensory fiber from here go to sympathetic ganglion, and from there fiber come back. So the three levels of such reflexes, as I mentioned previously, they can be through the enteric nervous system, which is local reflex, they can be through the sympathetic prevertebral ganglion, and they can involve the central nervous system also. When they involve the central nervous system, in this case gastroileal reflex, this will be vagovagal reflex. Am I clear? No question?
I do have a question about the sympathetic.
Okay, let me tell you. You want to know about sympathetic? Look, here are sensory fibers from stomach which are going through the vagus nerve. And there are also sensory fibers which are going through sympathetic nerves. Right? Usually students have a wrong concept that vagus is only motor. But actually vagus has sensory fibers also. And sympathetic nerves are not only motor, they do have their sensory fibers also. So the sensory fibers from here can go to sympathetic ganglion, prevertebral ganglion like celiac ganglion, or from GIT they can go to the superior mesenteric and inferior mesenteric ganglion. You understanding? From the ganglion, motor fiber come back to gastrointestinal system and they can alter the motility. How? Let me tell you. From sympathetic ganglion, the sympathetic fibers come here. If gastroileal reflex is increasing the ileal movement, then sympathetic supply to ileum will be inhibited. When sympathetic supply is inhibited, then inhibition of sympathetic nervous system on GIT is reduced and GIT become active. Right? You got me now? Yeah. Is that right? But, so what really happens in gastroileal reflex or any reflex which is through the ganglion, that if you are going to inhibit the target area, then you have to reduce the, sorry, if you have to increase the ileal movement, then ganglion should inhibit the sympathetic supply here. Am I clear? And secondly, these sympathetic fiber will go up to what? To the medulla through the vagus nerve, and in the medulla they will eventually stimulate the parasympathetic dorsal vagal nucleus, and those fibers which are coming to especially ileum, they are stimulated and they stimulate the enteric nervous system here, and again movement occur. Am I clear now? Okay, that's good. Now, after having said all these things, the chyme moves through small intestine at the rate of 1 centimeter per minute, approximately. It means in 1 hour, chyme move about 60 centimeters per hour, right, within the small gut. Now, after having said all of these things, we will come to another type of movement which also occur there. We have already discussed the mixing movement. We have already discussed the propulsive movement. Now again we talk about MMC. What are these? Some people call it migratory myoelectric complexes. Migratory myoelectric complexes. And there are other people who term the same thing as migratory motor complexes. So it's up to you what you want to call, right? Migratory motor complexes. Again, in previous lecture I told you such movement are generated in the stomach also, right? So MMC movement are produced in the stomach also during fasting, and these movements are also produced in gallbladder and small intestine during fasting, right? But the point to remember is MMC movements are not generated in large gut. So they are feature of stomach and small gut. So what MMC movements are doing here? MMC movement or migratory motor complexes, right? They move indigestible material rapidly through the small intestine. Is that right? For example, during fasting, whatever you could digest and absorb, you have done. And now the material which is left, residual material which is left, that should not be left there. Let's push it forward and dump into those areas where it should go to large gut and eventually become fecal matter, right? So what really happens that indigestible or non-digested material by the MMC during the fasting time is rapidly propelled toward the large gut. Is that right? And during the second part of the MMC, secretions from GIT help to dislodge such material from the walls of gastrointestinal system so we can clear the small gut from such contents. Any question here? No? Now we come to again another movement which can be produced in small gut is peristaltic rush. Right? That is peristaltic rush. Yeah, this is rush hour at gut. Right? Peristaltic rush is produced when there is strong irritation or there is very strong distension in some part of the small intestine. When small intestine really want to relieve itself, even though normally it works well with these movements, mixing, propulsive and MMC. But if small intestine is irritated too much or is distended too much and it wants to relieve itself, it will produce very powerful and very rapidly moving, very powerful and very rapidly moving multiple peristalses which will rapidly move the contents of small gut into large gut. Is that right? And this classical example of this is in infectious diarrhea. In infectious diarrhea, when the virus here or there are bacterial infections and they're irritating the small gut mucosa, is that right? That may lead to production of peristaltic rush, and these very strong movements, right, they do produce some central abdominal pain also, you see, colicky pain as these movements go down and push the content into large gut so that they try to relieve themselves from the presence of irritative or overly distending substances. So this is one way how small gut relieves itself, right? Any question here? Okay, this was something about the small gut movement. So can you repeat it? What are the small gut movements? Classically, they are mixing movement like segmentation. Yes. There are a propulsive movement which are peristalsis. Very good. And then there are, yes, MMC to clean the gut, right, during fasting especially. And then when there is undue irritation to small gut, it may produce peristaltic rush to evacuate its contents rapidly into large gut, right? After discussing small intestinal movement, let's come to the motility in colon and beyond. Right. So now we will talk about the movements of the large intestine in the colon and beyond. Right. First of all, we should know that in the colon or large intestine, the proximal half of it is concerned with the absorption. Mainly it absorbs the water and electrolytes. Right. And distal half is concerned with the storage of the fecal matter. Right. And elimination of the fecal matter, that is defecation. Now, these are the two primary functions, and during these, when the chyme is, look, this is small gut, and here is ileocecal valve, and here is ileocecal sphincter. Functional sphincter, which is, you can say, slightly thickened smooth muscle layer, and don't forget you do have appendix here. Right? Now, large gut, primary function is to absorption of water and electrolyte in the especially its initial half, and distal half, mainly it is the storage and elimination of fecal matter. And during this process, when the chyme enters to the cecum, and very slowly it goes through the large intestine to the sigmoid colon, it converts from semi-solid material to the fecal matter, right? And the movement of large intestine are most slow here, right? What I mean by most slow, that it will take 1 to 2 days for the material to reach from cecum up to the last part of the large intestine. But before I move forward, I remember one thing. You know, in last segment, I was teaching that there, what was that? MMCs. MMCs are feature of stomach and feature of small intestine, but they are not the feature of large intestine. Number one. Secondly, when MMCs occur in small intestine, I forgot to mention there that MMC strongly stimulate the gallbladder and help to clear the or empty the biliary ducts also. Is that right? So please add it there. Now we come to back. Before I really go into detail of movement of large gut, I will talk about ileocecal valve and sphincter. Right? Ileocecal valve is a small part of the ileum with its mucosa protruding into cecum. And proximal to that, there is a thickened ring of smooth muscle which act as ileocecal functional sphincter. Now, ileocecal sphincter and ileocecal valve, they are regulated very well, especially by the cecum. Is that right? Normally, they remain tightly closed, ileocecal valve, and they only work one way from ileum to the cecum, because cecum is highly, you can say, populated with bacteria. But in the ileum, it is sterile or bacterial population is very low. So one of the major function of ileocecal valve is to prevent the highly bacterial population from the cecum to the small gut, right? Secondly, ileocecal sphincter is very properly regulated, right? For example, when content in the cecum become more, ileocecal valve should become tight. Because if pressure in the cecum is increasing, there is a danger of spilling of large intestinal gut into ileum. So actually, when pressure in the cecum increases, the ileocecal valve and sphincter tightly close. Right? We can say it's a type of ceco-ileal reflex. Right? Of course, this should be ceco-ileal reflex. Should be maintained through local enteric reflexes also, plus it is also maintained through the sympathetic prevertebral sympathetic ganglion, right? That fibers from the sensory fibers from the cecum will go and sympathetic ganglion, through the sympathetic ganglion, fibers will come back to the, what is this? Ileocecal sphincter and tighten it up. Is that right? But gastroileal reflex opens it. When you take a decent meal, right, then what happens? There is gastro from stomach to the ileocecal valve. Right? So gastroileal reflex, that when you are taking meal and your stomach is stretching, ileum is activated, right? That actually also relaxes this valve so that content of the small gut go to the large gut, and so that small gut can accommodate the incoming chyme from the stomach. Is that right? So gastroileal reflexes opening the valve. Is that right? And now we come to large gut after having discussed this. In the large gut, let's talk about the type of movements. Number one, there are special type of mixing movements here, and which are called hostrations. Hostrations. Right? What are hostrations? Hostrations are that, let me draw here more appropriately. Hostrations are the appearance of constriction at smooth muscle contractions, right? Smooth muscles contract, right, at intervals in the large gut. And when those smooth muscle in muscularis externa contract, they make the lumen narrow. And in between the two contractions, wall of the colon bulges out like a bag, swollen bag. Is that right? Actually, two processes occur here. Number one, smooth muscle contract. Is that right? And make it what? Circular constriction. Secondly, tinea coli also contract and shorten the large gut. So when large gut segment becomes shortened and at multiple constriction ring appear, right, then large gut areas in between the constriction ring will convert like swollen bags. These bags are called hostra. These bags are called hostra, and such movements are called hostration. So what are hostrations? These are movements in which at almost regular interval, smooth muscles of large gut constrict and tinea coli also constrict. When smooth muscle constrict, they reduce the luminal diameter and produce the constriction rings, and when large gut tinea coli contract, they shorten that segment. Due to this reason, large intestinal content, right, are compressed in between these constriction, and this act as a mixing movement, and like this these constriction dug into the, you can say, semi-solid material there and chop it again and again, because hostrations appear and then again appear, then after some time again appear. You are getting it? So at different sites. So in this way they bring different areas of semi-solid content of large gut to exposure to the where? To the mucosa, so that water and electrolytes can be absorbed, and gradually this semi-fluid content of large gut convert into semi-solid fecal matter. Am I clear? Right? So these bags are called hostra. H-A-U, hostra. And these movements, mixing movements which produce hostra, they are called hostrations. Any question up to here? Right? Then another thing, while these hostrations are produced, there can be very weak propulsion also. There can be very weak propulsion, and content may spill from one hostra to the next hostra. Is that right? And these very weak movement, which I can push the content from one bag of hostra to the next bag of hostra, these movements are called segmental propulsions. So there are segmental sacculations. These are hostrations. And there are segmental propulsions. Let me write it here. Mixing movements are, number one, segmental. What is this? Segmental hostra production. That is hostrations. Second movement can be segmental, yes? Propulsions. These are very weak. They are not very strong, and they just push content from one hostra to the next. Is that right? That is called segmental propulsions, right? Then there can be very important movement. If you forget all of the things, I don't care. But now what I'm going to tell you, you must remember. That is the mass movement. Mass movement actually occurs in different people with different frequency per day, right? Usually in some people, mass movement occur 1 to 3 times in a day, and in other people, mass movement may occur up to 10 times per day. Now, mass movement are very important. What are mass movement, right? Mass movement, let me tell you how a mass movement is produced. A strong constriction ring is produced, let's suppose here. Very strong constriction ring is produced, and this area becomes very narrow. And then distal to it, for about 20 centimeter, hostrations disappear. Hostrations disappear for this segment. And then look, all these hostrations distal to the ring disappear. Suppose this area has become now very narrow due to this very powerful constriction ring. And distal to area hostration disappear, but look here, all of it like one tube from all side compresses, right? So all of it actually become compressed. And when all of it become compressed, what it will do? The contents which are present in this about 15 to 20 centimeter of the lumen, they will be pushed en masse distally. Again, let me repeat. What are mass movement? Let me put it here. It has nothing to do with mass. Okay, let me explain what is mass movement. Now, in mass movement, different people develop at different frequencies per day, but usually they come after many hours. Most commonly, they appear within 1 hour of breakfast. You know, that is what leads many people to the defecation, right? Within 1 hour of taking breakfast, mass movement may appear 10 to 20 minutes, right? What are mass movement? But they may occur in other times of the day also. What are mass movement? Mass movements are a very strong constriction ring appear in a part of a colon, and then about 20 centimeter distal to that area, hostrations disappear, and all that area contract as one unit and push the content en mass as one mass distally. And then mass movement may appear here and push the content forward. And if multiple mass movement come and lot of contents are pushed into rectum, desire for defecation will be there. Because normally rectum is kept empty. It's a wrong, some students have a wrong concept that rectum is all the time full. No. Usually, most of the day and night, rectum is kept empty. Why? Because contents of residual, you can say, material or the fecal matter is in the second half of the colon. But number one, there's a weak functional thickening of smooth muscle at recto-sigmoidal junction. That prevents the overflow of material into rectum. Secondly, there's acute angulation here. That also prevents overfilling of the rectum. So normally, rectum is almost empty. Is that right? But when multiple mass movements are generated, they push the material to the rectum. That produces the desire for defecation. Am I clear? And these mass movements, once they start, usually they persist for 10 to 30 minutes. They keep on reminding that if you need to go to for bathroom for defecation, right, or anywhere for the defecation, it depends on your choice. Now we come to another thing, that mass movement can be initiated by gastrocolic reflex. You know, many people when they take a heavy meal and within a short time they need to go to washroom for defecation. Why? Because there may be gastrocolic reflex. Again, this gastrocolic reflex may be coming through, yes, sympathetic ganglion, or it may be coming through, yes, right? It may be coming through central system, right? So what is there? The gastrocolic reflex stimulated. Plus, it can also be initiated by duodenocolic reflex, right? To further enhance mass movement, because when duodenum is also stretched, it means you have really eaten a lot. So you need to evacuate at the end. Any question up to this? No question? So what are mass movements? These are high amplitude movements and they are propagating contractions, right? Now we come to defecation reflex, that how the defecation reflex work. Is that right? So we have talked about the mixing movement in large gut, which are hostrations. We have talked about segmental propulsion, which are very weak propulsive movement. And then we have talked about the mass movement, which are the most important to remember, which occur in the colon and which can push a large amount of colon luminal contents and mass distally. And if they push the material up to the rectum, that may initiate the desire for. Now we come to the defecation reflex. Let's understand it with clarity, because this is very important to understand. But before really I go into defecation reflex, I would like to ask that normally, of course, healthy person does not have dribbling. Healthy person who has gone through toilet training, and including the children who have been toilet trained, they don't keep on dribbling their fecal matter. how the dribbling of fecal matter is prevented? Normally, how the dribbling of fecal matter is prevented? You don't want to tell me? It's no secret. Most of the doctors know. Yes. Yes. That is simply by the internal and external anal sphincters. Internal anal sphincters is here, right? And this is a ring of smooth muscles. Actually, in the muscularis externa, the smooth muscle layer becomes very thick as it reaches, what is this? Anus. Secondly, I will give a little different color, and outer to this, there is skeletal muscle, which act as what? External anal sphincter, right? So what we can say, there is internal anal sphincter and there is external anal sphincter. Let me draw it again. This is internal anal, right? And here we have overlapping it external anal sphincter, right? Now, internal anal sphincter is involuntary. It's part of smooth muscle. And external anal sphincter, thank God, can be controlled voluntarily, right? Now, and there is a role of what? There is a role of central nervous system also into this situation. So let me draw it. Right. Now, what really happens, how the defecation reflex is initiated. Actually, as I told you previously, normally rectum is kept empty. Is that right? But whenever rectum will become full and rectal mucosa is stimulated, desire for the defecation is initiated. Normally what happens, before the desire for defecation start, the multiple mass movement. As I told you, mass movement can be initiated by the what? Gastrocolic reflex, or by the duodenocolic reflex, or spontaneously few times a day. Once the mass movement are there, and if there are enough fecal content over there, right, then this fecal content will be pushed through the descending colon and sigmoid colon into rectum. And when rectum receives these contents, when the rectum receives these fecal contents, right, these are the contents which are pushed forward, right? When rectum receives these contents and there is rectal stretching and irritation to rectal mucosa, number one, local reflex, my enteric reflex is stimulated. Is that right? What is that, my enteric reflex? Actually, the local sensory system stimulates the ascending pathway, and this ascending my enteric system stimulates all the way up. It stimulates all the way up. And this stimulation creates more mass movement. Is that right? So it means once the rectum is slightly stretched, right, this will, what it will do, through the myenteric reflexes, the ascending pathway will stimulate the transverse colon, descending colon, and of course sigmoid colon, and all of them become stimulated. And when all of them are stimulated, that will lead to movement of more fecal material here. And at the same time, look here, not only an ascending pathway that it stimulates, suppose this was the sensory sensations from this area. Ascendingly they stimulate, but descendingly, especially internal anal sphincter, they inhibit. Of course, by releasing the VIP and nitric oxide, right? So, what you have to remember, first initial mass movement, filling of the rectum, right? Rectal stimulation actually stimulates the myenteric plexus strongly, right? And ascending pathway stimulate the, what is this? More proximal part of colon, and more mass movement bring more fecal material here. At the same time, what is there? Relaxation of internal anal sphincter. Is that right? Secondly, this intrinsic defecation reflex, this is intrinsic reflex because no other outer part of nervous system has been involved. So what should be this reflex called? Yes, this reflex should be called intrinsic defecation reflex or intrinsic myenteric. To remember it more clearly, intrinsic myenteric defecation reflex. Usually this intrinsic myenteric reflex is weak. And remember one thing, as soon as the smooth muscle in the internal anal sphincter relaxes, if we are not ready for defecation, we actually consciously can contract external anal sphincter. You are understanding?
So it's not inhibiting because you have a mind.
No, look. This is very important to understand. When fecal matter come here, so sensations in the rectal mucosa stimulate the myenteric plexus proximally as well as distally. It stimulates the proximally and inhibits distally. When it stimulates proximally, more mass movement bring more material, and distally it inhibits the internal anal sphincter. Thank God, again, we have external anal sphincter. Actually, we reflexly close it. If we are at some inappropriate situation, you are watching a video and you cannot go rush to the washroom, what you will do? That you will keep your external anal sphincter tight, right? This is one thing. Secondly, when more material come, right, this weak reflex can be amplified or intensified by parasympathetic reflex. How? Look here. These, some of the sensory fibers, from where? What is this? Rectal stretch area mucosa, they go to the spinal cord, lower part, into conus medullaris as 2, 3 and 4. Right? And from here, parasympathetic fibers come through nervi argenti or pelvic nerve, and these parasympathetic fibers, look now, they stimulate what? Listen. Fibers from here, sensory fiber went to sacral part of spinal cord. From here outflow come. This outflow further inhibit what? Internal anal sphincter, and this outflow further stimulates what? Myenteric intrinsic reflex. It intensifies that. You know, when parasympathetic stimulation comes, myenteric system become amplified. So what really happens, that intrinsic myenteric reflex for defecation system is weak, right? And what really happens, along with this intrinsic, rectal stimulation with more incoming fecal matter stimulates the parasympathetic outflow from sacral segments, and through the pelvic nerves parasympathetic outflow comes and it further stimulates the myenteric system, right? And the result is that intrinsic reflex become more intensified or amplified. So there are more strong and more stronger which movement? Mass movement. Along with that, more stronger relaxation of what? Internal sphincter. But if you really don't want to go to washroom and it's not nearby, what you do? You keep your external anal sphincter now consciously tight. Is that right? Until if you remain like this, what will happen? You know, sensory system will think that you are not responding, and when you don't respond to someone, what happens? Response goes down. Same thing happen here. When you are not responding to this defecation call, intrinsic defecation reflex, amplified by, what is this? Parasympathetic defecation reflex. If you are not responding to that and keeping it tight for a while, then rectal sensory fibers will get used to this high pressure. So for a while this reflex will die out. But after some time, depending upon how urgent is the situation, 10 minutes, 20 minutes, 1 hour, 2 hours, again the call will come. But let's suppose this time, you just make it tight and you go to the washroom. And if you reach there, now you are comfortably sitting there, right? And now you really want to defecate. What you will do? One thing is, this decision, conscious decision to make defecation is done at the higher center. Right? You really think about it, you should defecate. So what will happen, that from higher centers, fibers will come down and they will control the outflow of what are these fibers? This is pudendal nerve. What is this? Pudendal nerve which control the external anal sphincter. So what you do, that you inhibit the outflow through pudendal nerve, and external anal sphincter also relaxes. Is that right? So internal anal sphincter was already relaxed. But sometimes it happens, by the time you go to washroom, right, the desire for defecation has been reduced, or we can say desire is there, but this pathway, look, this whole system is reduced. So what you can do, you can amplify it by conscious effort. So you take a deep breath, close your glottis, and then what you do, contract your abdominal muscles and diaphragm. What you are doing? You are raising the intra-abdominal pressure, right? Actually, you are squeezing the whole colon to push more material there, so that this pathway is again stimulated and parasympathetic system is also stimulated. You understand it? Valsalva maneuver, right? Especially if you feel that you are in rush to do defecation, and maybe on 2 minutes you have to be out, or someone is knocking at the door. Yes, there are so many situations. Or you are a person who want to save time everywhere. So under those circumstances, what you do, you close your glottis, right? Contract abdominal muscles and diaphragm, right? Of course, diaphragm downward, abdominal muscle inward, and increase intra-abdominal pressure. At the same time, you relax the muscles of pelvic floor. When muscles of pelvic floor is relaxed, right, that also help the external anal sphincter to come outward, right? And at the same time, you have inhibited consciously, what is this? Pudendal nerve, and external anal sphincter become relaxed. When internal anal sphincter is relaxed, at the same time external anal sphincter is relaxed, at the top you are pressing the whole system like that, right? Then what will happen, that this reflex, intrinsic, what intrinsic? Intrinsic defecation reflex with parasympathetic defecation reflex, which are automatic, they become further amplified and they move the content outward and defecation start. Am I clear? So what are the 3 important steps you have to remember? What are the important steps for defecation? Listen. Number 1 is mass movements. Mass movement. Number 2, filling of rectum. Number 3, yes, desire for defecation. Desire for defecation. And at the same time, 3A is desire and 3B is, as soon as rectum is filled, they stimulate what? Intrinsic myenteric reflex. Intrinsic myenteric reflex, right? Which increases the movement and internal anal sphincter relaxes. But if situation is inappropriate, right? If the situation is inappropriate socially, it is not acceptable to defecate whatever you are doing, then what you will do? You will contract external anal sphincter, and gradually that reflex will die out. But if it is, you go to washroom and you are comfortable and you decide really to go for defecation, what will you do? The next step should be that you will do the Valsalva maneuver. What is that? That basically you will take a deep breath in. What are the components of this? Take a deep breath in, right? Close the glottis. Then contract abdominal muscles and diaphragm. Raise intra-abdominal pressure, so that colon contents can be pushed forward further, right? Plus relax, what is that? Pelvic floor muscles, right? And once you have initiated the Valsalva maneuver, at the same time you inhibit, what is that? Inhibit external anal sphincter, and defecation should occur hopefully. Is that right? Any question here? There's no question. Now, few important things about the disorders of GIT motility, right? Just few words I will talk about these disorders of the GIT motility. Number 1, ileus. Paralytic ileus. Paralytic ileus is when the whole myenteric plexus in GIT becomes paralyzed. This type of situation is seen after general anesthesia, or it is seen when abdominal surgery and you manually handle the gastrointestinal system, or when there is severe electrolyte imbalance, right? So, or there is septicemia, or there is peritonitis. So anything which generally irritate the GIT that may lead to paralysis of enteric plexus and gastrointestinal movement stop. That is called ileus, right? The classical example is general anesthesia and post-operatively, but after some time it recovers. Is that right? Then this is one condition. Second condition is Hirschsprung's disease. In this disease, this is usually congenital condition. What happens? One segment in the large intestine does not have ganglion and neuronal cell bodies. You know that these in enteric plexus and submucosal plexus, both of them, they get neuronal cell bodies or ganglia from the migration of neural crest cells. Sometimes neural crest cells don't migrate properly, and in some segment of the large intestine, ganglia will not be there. We say that there's a ganglionic segment. And when there's a ganglionic segment, what happens? Let me explain. This is congenital cause of megacolon. But these ganglion can be destroyed by a parasite called trypanosoma cruzi. That can also destroy these ganglion, and that can produce also similar megacolon type situation. But classically, what happens in these cases, let's suppose this is large gut. Suppose this segment does not have ganglion. If this segment does not have ganglion, then what will happen? All the food matter which is coming here, it will accumulate proximal to that, because this will remain semi-contracted. Why it remains semi-contracted aganglionic area? Because there is failure of relaxation. You know, when peristalsis will bring the food content here, normally more distal area to the peristaltic ring should relax. But because there are no ganglia here and no neurons here, enteric system, so peristalsis are bringing, or you can say propulsive movements are bringing a lot of content in the lumen of GIT just proximal to the aganglionic segment. But aganglionic segment itself cannot generate relaxation, right? So it does not receive and accommodate incoming contents. So what really happens in these patients, this part of the colon become very much dilated, proximal to that. That is why this is also called megacolon. The treatment of such situation is, of course, if unfortunately baby is born with this, it's more common in male babies. What will happen? There will be delay in passage of meconium. Plus, because contents in GIT are retained, and then baby starts throwing up, and there's nausea and vomiting and all those things. Treatment of this is a surgical resection of aganglionic segment. Am I clear? Then another condition which is there is irritable bowel syndrome. This is very common condition. Irritable, don't confuse it with inflammatory bowel disease. That is entirely a different category of disease. I'm talking about irritable bowel syndrome. In this syndrome, the patient suffers with abdominal pain, vague abdominal pain, and with that, so they repeatedly suffer with dysfunction of GIT motility. Sometimes they develop diarrhea, sometimes they develop constipation, right? And this irritable bowel syndrome is now thought to be due to increased visceral hypersensitivity. That in gastrointestinal system, the sensory neurons which are supposed to move the enteric plexus, they are oversensitive. Is that right? So they are oversensitive. So visceral hypersensitivity is there, and that leads to exaggerated intestinal reflexes. Right? And due to that reason, these people may have repeated diarrheas, right, or abdominal pain when the exaggerated movement in the GIT. Clear? Then last but not the least is fecal incontinence. There can be many reasons for fecal incontinence, right? But more common are when these neurons, what are these neurons? Parasympathetic, what is this? Neurons which control the external anal sphincter, right? They are disrupted, right? Or higher control to the sacral area is interrupted. For example, if you have a lesion in spinal cord above the conus medullaris, right, and higher control cannot come down. Now what happens that if there is no higher control, the function of this higher control on lower spinal reflexes was that when socially it is not acceptable you should defecate, what you do? You inhibit the defecation reflexes. And when you are in the washroom and you want to defecate, consciously you can slightly assist the defecation process by producing Valsalva maneuver and relaxing the external anal sphincter. Is that right? When this this is lost, what really happens after some days, all the systems start overfiring. And if conus medullaris itself is functional, just it has lost its higher control, then what happens? These reflexes, they become automatic. Am I clear? And those automatic feature, right, that whenever fecal matter is reaching here, right, it will stimulate, first of all, intrinsic reflex, then stimulate the parasympathetic reflex. But if in lower motor neurons, if external anal sphincters, these fibers are overfiring, this may lead to retention. Is that right? Usually it leads to incontinence. But if pudendal nerve is overfiring, you remember that some of the lower motor neurons, when they are no more controlled by the upper motor neuron, they start overfiring. If it's overfiring, there will be some degree of what? Retention of fecal matter. But giving a little enema here, right, every morning, you can initiate the defecation reflex and get the stuff out. Is that right? Any question up to this? I'm talking about when lesion is in the nervous system above the conus medullaris. So conus medullaris, so sacral area of the spinal cord is functional, but it is no more controlled by the descending pathways. So consciously we cannot alter or override the spinal reflex. Under these circumstances, that becomes a semi-automatic system. Am I clear? In that case, what really happens if visceral, this intrinsic reflex and parasympathetic reflex is very strong, whenever fecal matter reaches here, person will automatically evacuate. But if external pudendal nerve is very strong, then it will lead to retention. You are getting it? Under those circumstances, we can give enema in the morning. Usually we do to these patients after the food. So what happens? Why after the food? We are taking the help of gastrocolic reflex. Plus, that enema will irritate this area and distend this area, and these reflexes will become powerful enough to override the external anal sphincter and get the stuff out. Is that right? Any question here? Okay, class dismissed.